I hope you found the discussions and presentations over the past 2 days valuable. As you can find in your program schedule, we have talks over here as well as the oral presentation running parallel on the upper floor. We'll now begin with session 11. For this, I would like to invite Dr. Janki Ram Vaitla and Dr. Shelly Rana to chair the session.
Good morning, all of you. Last 2 days we have a fantastic lectures. We have seen many. So today for the session 11, I would like to introduce the first speaker, Dr. Sandeep Haldar. So I'd like to give a brief introduction about Dr. Sandeepan Haldar. So Dr. Sandeepan Haldar completed his PhD in chemistry from the Indian Institute of Technology, Kanpur. His first postdoctoral research at Bar Ilan University, Ramat Gan, Israel, focused on physical organic chemistry. He was later awarded the prestigious India-USA Fulbright Nehru Postdoctoral Fellowship by the United States India Educational Foundation, USIEF, to pursue his research in asymmetric catalysis for hydrogenation reactions at the Ohio State University, USA. Upon returning to India, he joined NIT Jamshedpur before moving to Visvesvaraya National Institute of Technology, Nagpur, as an assistant professor in 2016. Dr. Haldar has been awarded research grants, including the Early Career Research Award from the SERB Government of India and Indo-Russian and Indo-Philippines Collaborative Research Grants from DST Government of India. His research group is currently working on organocatalysis, particularly hydrogen bond donor catalysis, with applications in CO2 fixation in organic molecules and the stereoselective synthesis of heterocycles. So today's title of the talk is Hydrogen Bond Donor Catalyzed Domino Transformations, an efficient tool for the synthesis of biologically prevalent ring system. With this short introduction, I would like to invite Dr. Sandeepan Haldar on the dais.
Thank you for the kind introductions. Good morning, all of you. First of all, I should thanks to the organizing committee, Professor J.S. Jadhav and Professor Vishnu Maya, because my presence was not confirmed earlier, but still they have accommodated me in these sessions. I'm grateful for them. So as you can see, the title of my today's talk is Hydrogen Bond Donor Catalyzed Domino Transformations, an efficient tool for the synthesis of biologically prevalent ring systems. So I'll take you in a part of two different aspects. One is the domino protocol. As you can know that if you start with some process where the initiation is needed, and after that the things are going on automatically. So in terms of chemistry, once the synthesis starts, you just put the substrates with the reagents, and then one after another the sequential steps will be going on without addition of some other reagents or isolation of the intermediates of the product. This is related to the dominoes, and the other part of it, it's hydrogen bond donor catalysis. As you can know that hydrogen bonding is basically a stabilizing factor, and which is common in DNA and in other naturally occurring things. But including this particular type of stabilizing factor in catalysis, that is a challenge. Once the substrate will form the hydrogen bond with a particular type of orientations, and that will be catalyze the reactions for the further transformations. So utilizing this concept, there are having different kinds of hydrogen bond donor catalyst based on their mode of forming the hydrogen bond. This is a single hydrogen bond donor catalyst. It might be a dual hydrogen bond donor, or it might be a bifunctional hydrogen bond donor, where the hydrogen will be forming the hydrogen bonding, and there will be some other part which might be a Lewis acid center or creating some chiral inductions on the substrates. So with that, these are some reported catalysts where you can see even the vitamin C can also form hydrogen bond to a substrate and catalyze the reaction. So this way we understood that which particular type of hydrogens will be required in order to make the hydrogen bonds, where from those hydrogens will be coming, and what are the other aspects are needed in order to design a new catalyst which might be a hydrogen bond donor catalyst. So in terms of understanding these sort of aspects, we figure out that it should be our non-covalent interactions to accessible hydrogens. there would be some like the suitable heteroatom in order to attach with the hydrogen to that, and it should be very much rigid structure, otherwise the bond rotations or planar structures will be needed in terms of making the hydrogen bond very fruitful. So with that, we have designed some very simple structures, and we are hoping that these colored hydrogens will be forming the hydrogen bond with the substrate and in order to catalyze the reactions. With that, these are synthetic steps, is very common, the condensation, nominal condensation kind of reactions. We have chosen some 1,3-dicarbonyl compounds and putting the hydrogen in a proper place, and accordingly we have designed and synthesized these compounds. To start with, we had a project on that carbon dioxide installation. So we did the reactions with epoxide and carbon dioxide in the presence of the catalyst, and we have found this catalyst is working pretty well over there in these particular reactions, but compared to the others, they are not. So we are hoping that here the hydrogen bonding is more proper, more suitable, which is stabilizing the substrate and making the further steps, because it's a domino process. And then then we have generalized the method, and like in terms of the substrate scope also, we have found all the cases the products are coming in good yields and the conversions. Later on, we realized that what are the main criteria to form the hydrogen, because once the reaction goes with this catalyst, we are very happy the reaction is going, but we are more happy when the reaction was not going with this catalyst, because that is proved that this distance matter in order to coordinate to the substrate, then only the reaction will be catalyzing. So in our this library, this is the best catalyst, and these, by changing one positions, we have just changed this phenolic OH from here in the other positions, reaction is not been catalyzed. So that's finding to have for the further proceedings in this particular area. And then here we have seen that that might be the hydrogen bond, but because of that single bond rotations, this is also not catalyzing the reactions. So in order to move forward, we wanted to utilize further of our catalytic method, and we have targeted this sort of isoxazolinone kind of products, because this is very common in different kind of biologically important compounds, including drugs and all. So accordingly, we have chosen the substrate with epoxide and this isocyanate electrophile. And to move on, here also we have found the corresponding products in very good yields, and most importantly, it is completely atom economic, and there is no other products are forming in these particular reactions. Furthermore, we have basically gone for the generalizations of different kinds of isoxazolinone in this case, and then we move towards the asymmetric versions. We have incorporated the starting material, which is enantiomer, and followed by you have moved with this particular domino reactions and found the products are coming as an enantiomer product, which is a single enantiomer is forming in these particular cases. That means the reactions are occurring from a particular side, that because there is a possibility to open this epoxide from this side or the other side, we are getting a single regioisomer in this particular case, and reaction is completely going in an enantioselective product. With that, further moving, we have taken different kinds of electrophile in terms of fixing them with epoxide in our catalytic method to have a particular functionalities in the products. So to do that, we have further taken this malononitrile derived electrophile, which is very much activated electrophiles, and we found here the reaction is going on. Though both the diastereomers are forming, the astroselectivity is about 90:10, about 80% DE. Cis is the major product, and the trans is also forming. So what we have done, we have separated the diastereomer by column chromatography and separately crystallized them, and to understand their structures conformed with that. Further, we have generalized, we have incorporated different kind of like substituents in both the cases from epoxide as well as from the electrophile in order to make a variety of tetraidrofuran 2,5 disubstituted. All the cases, the cis products are coming as major. Few cases, the diastereoselectivity is very high, and some cases, the trans diastereomer is also forming. Then we have gone for the inductions of stereochemistry here. Here also we have found the reaction is going very much well for the enantioselective, like the chiral epoxides, and then get the corresponding products as a single enantiomer in this particular case. And here also the reaction is completely regioselectives in nature. So in terms of making the proposing the reactions, we have found that initially this sort of hydrogen bond on a catalyst is coordinating, making the hydrogen bond with the substrate epoxides, followed by the co-catalyst, whatever you have used, that tetra-butyl ammonium iodide or this sort of alkyl iodides, quaternary ammonium salts. and then followed another way. This might be opening from the other side to get the other isomers, but which is not at all forming. So it is moving on a particular directions to get the products in a single isomers. And other case, there's a diastereomer is forming, but very less in amount. So this particular work we have published in JOC, and this is a cover page also we have accepted. Here we have proposed that these two ideas which are coming from the two different electrophiles which are hitting to the substrate containing the catalyst, and then the reverse are coming from there in different form, this one, and products are floating with that. So they accepted this particular type of concept and accepted as a cover page in JOC. Furthermore, we have to utilize our synthesized compound tetradofuran for to make some different kind of varieties of the products. In our case, the main substituents which we can move forward, there is the true geminal cyanide groups, which are quaternary carbon center, which are present over there. So we initially targeted these sort of products, which is coming and aiming to synthesize from there. And another one is another heterocycles like the fused furo 3,2,C quinoline. Now, why is it so? Because this is the core of a marketed drug that is called avibactam. That is the non-beta-lactamase inhibitors. Here, the core is the diazobicyclooctane, which we are aiming to synthesize from our already synthesized tetradofuran. And accordingly, we have designed and teaching over there. So beginning to that, we have designed something and did some in silico studies to get that whether exactly our design compounds can bind and can have that. And these are design compounds that mostly it is coming, though these values are not very much or in silico studies only on computational basis. And this is the avibactam, which is the drugs that antibiotic drugs. So accordingly, we have started, we have designed. So initially what we have done, we have targeted to reduce these two geminal cyanide present in the quaternary carbon centers, followed by we will be doing the next step. But once we are starting the reductions, we have observed that we are not getting both the cyanide reduced. It is reducing only single cyanide over there, and which is anti to the substituent which is present over there. And in order to do that, further we have targeted to do the Mitsunobu cyclization, but it was not going. But after that, we have undergone the cesium carbonate, and here the leaving group is also changed, and we could get that breached compound. This is also characterized by single crystal XRD. It is forming by two steps after getting the tetradofuran, then reductions with that single cyanide into that, and then further cesium carbonate mediated intermolecular happen to get these 5-6 breached tetradofuran one side and that piperidine in the other side to reach that core, and then further we'll be proceeding for to get that diazobicyclooctane. That part is left. So we'll be coming to a variety of these kind of compounds which will be mimicking to that avibactam structure to get their corresponding biological activity. So this is not yet published, few works are left as well. Now we are targeting another heterocycles, which is also 3,2-C furo 3,2-C quinolines, which are very much important pharmacophore found in different sort of biological activities, anti-cancer properties. They are the different kind of like that enzyme inhibitors and this way. So we are targeting this particular type of heterocycles from our synthesized tetrahydrofurans. In order to do that, we have gone through the literature, but there are very few reports known where this sort of furan and the fused with that quinolines are reported, and these kind of things are mostly synthesizing from the furan only. So where our tetradofuran is already synthesized, we have to derivatize the other functionality and to reach up to these sort of fused heterocycles. So in order to do that, what we have done, again we have started from the reduced product and just to do some metal catalyzed CN coupling. But here in every cases, we have to keep our bromine at the alpha positions in order to get that core. And here also we got the products in the copper mediated copper iodide catalyzed reactions. But to do that, every cases we have to keep the bromine in the ortho positions to get this this particular type of cyclization. So that's actually restricted the substrate scope of it, so that we cannot vary the these sites. In order to find some alternate ways, we introduce the hypermanent iodine reagents in order to go for this kind of particular CN bond formation. But once we have started this particular reduced product in this particular medium, in the presence of that hypervalent iodine, we found it is oxidized product is forming. It is reoxidizing. Whatever the primary amine is forming, that is reoxidized to the nitrile. And then we found that that is already reported in the literature, that this primary amine once you will be taking in the presence of hypervalent iodine, it will be reoxidizing to that particular cyanide. Then what we have done, we have just protected it with a tosyl, and that too also going for the further cyclizations. And here we have the corresponding products in this particular medium. And then we have just characterized this tosylated product by single crystal study and found this particular steps are going pretty good in terms of making the products after these two steps. But additionally, you have to do the tosylations followed by cyclizations. So then we have generalized the protocols to have all different kind of varieties of these tricyclic furo 3,2C quinoline kind of structures. And this is the proposed mechanisms. This is normal hypervalent iodine mediated reactions. I'm not going to do the details of it. And after that, I'll take you to one more domain. Here, actually, initially you have started with normal epoxides. Now we wanted to introduce the cyanide, which is also synthesized from that our electrophiles, and in order to understand that reactivity. So to go for the literature search, we have found that this particular type of epoxide is highly reactive. It can open from the CC bond, it can also open from the CO bond, depending upon the reagent systems in the microwave reagents and all. And this particular type of epoxide we have actually synthesized from our electrophiles. So furthermore, we've gone through some more literature on these particular things. Here also they have converted to the tetradofuran or a variety of different products. But most interestingly, here the byproduct which is forming, that is the cyanide is coming out from there. That is a hydrogen cyanide is coming out. So there is a concern of atom economy, that whatever the cyanide you are taking, you are unable to install them in the product. So you are losing the cyanide. And that too also cyanide releasing from the reaction is not good for the industrial purpose in other case. So in order to take these particular things in our mind, we kept our objective that what will be the fate of these reactions if you use them in our hydrogen bond donor catalyst. So in order to go for it, we are expecting maybe this sort of epoch, the tetradofurans will be forming, where this kind of two different quaternary carbon centers will be there in our tetradofuran, and in order to have the corresponding reaction. But to do that, we have isolated the products, that's a cyanotransfer reactions, and that too also cyanide is forming directly coupling with the sp2 center to tricyanide product, the tricarbonitrile products. Then we found this is not our desired product, or we have planned. Once we are keeping these sort of reactions in the one equivalent of that, and then we isolate the product, we characterized by NMR and all other single crystal XRD as well. And then we found these tricarbonitrile products are a good material for different kind of dyes. Their electrochemistry, their photoselective sensitivity, nonlinear optics, charge transfer complex could be used. So in one step, we can do that particular type of product. That was our initial understanding. But then we need to also understand what is the atom economy. Here, this part is coming, that cyanide is coming from here. But what about the rest of the, okay, 2 minutes. I'll just go through it. And then we moved for the to optimize these things, and we have gone through the different kinds of derivatization of it to variety of these tricarbonitrile products we have synthesized in terms of doing this particular way. Here we have found that the ascorbic acid is working better as compared to our synthesized hydrogen bond donor catalyst. And furthermore, we have just variety of it. We expected that this will be the other product which will be coming from here, but actually this is not. The other product is coming, which is another pyran kind of derivatives from here, and that too, here it is one equivalent and one equivalent. The complete atom economy is there, but very poor yield. Then we move on, that is also characterized by single crystal XRD. Then we have used two equivalent of the electrophile in order to get these two products in a single step. So here we have optimized the reaction in this particular our hydrogen bond donor catalytic methods, and we found both the products are forming in pretty good yield with complete maintaining of the atomic economy in this particular case. So we have variety of the products we have synthesized in this particular domain, and then we have varied that cyanide to ester, and here also we didn't get this sort of corresponding pyran derivatives. Rather, we get the tricarbonitrile, and another one, this sort of cinnamate derivatives we have seen forming in this particular case. Here also the products have been characterized, and this is recently published, this particular product. So we have just varied the initial substrate from a normal epoxide to a highly activated geminal disulfide. The corresponding fate of the reaction is entirely changing. That's what our observation in the presence of our hydrogen bond donor catalyst. So now we have also studied the how the cyanide is coming, because this is a sensor which can have the color change in the presence of TC and Q, and accordingly we get the like expected spectral pattern to get the cyanide. Now I think I'd conclude with that to get that developed hydrogen bond on a catalytic method to get a different sort of oxy and aza-heterocycles with a fused ring system as well, which are having corresponding biological activities. And these are the funding agencies which has to be acknowledged. Without funding we cannot move forward with our research. So that SCRB, that DST for that bilateral Indo-Russian and Indo-Philippines project which are going on, and these are the students which are very much dedicated and very active in this particular research domain. And these are my some other collaborators who are working in different biological activities on the material and other. And these are my international collaborations which are associated with me for the project. And this is a like view of our institute campus in VNIT Nagpur. This is Nagpur is the center of India, so it's an orange city as well. You can have lot many oranges over there. And thank you very much for your kind attention. Thank you.
It's a very good presentation, Dr. Sandeepan, on hydrogen bond catalyzed transformations. So the session is open for questions. So only one or two questions.
Very nice talk. So I have a curiosity, your hydrogen bond donor catalysis.
Yes.
So you showed that on enol bond. What is
Yes.
the enol content of that catalyst? Because
Uh,
generally keto form
You
will
mean
be.
the tautomeric
Yeah,
ratio?
exactly,
Okay,
exactly.
so we have undergone that because once we do the NMR, this is always showing that enolic weight as well as phenolic weight. And that too is also like we are hoping that both the weights are needed to coordinate with that. But you are correct that we cannot track actually that how much is keto conversion.
Yeah.
But based on the NMR study, we don't see any kind of that ketone peak over there. Mostly it is enol and this one that we have confirmed.
Okay.
Even we have crystal structure also, it is showing that enolic weight is
Enol is
present
there.
over there. Yes.
Otherwise, on modification can be done if that is the kind of
Yes.
aromatic ring with ortho subship. Means Glyceraldehyde, if you do the imine formation with orthohydroxyamine, that is also going to give you the hydrogen bond.
Oh no, that will cyclize. We have tried. You mean to say that alpha, that
Yeah, exactly.
that beta amino ortho aminophenol and salicyldehyde, and that will cyclize. That will not be, hydrogen will not be there. It will be cyclizing. That is the already reported method.
Okay.
Yeah, we have tried. That is cyclizing. That is a bicyclic things will be happening.
Okay. Okay. Thank you.
Okay. Next presentation from Sandeepan. So we hope that on that epoxide you can further expand much.
Yes.
Okay. Thank you. Thank you very much.
I request Dr. Jankiram Vaitla to felicitate Dr. Sandeepan Anhaldar. Sandeepan Haldar.
Okay. So now we'll introduce second speaker for this session. So the second speaker is Dr. Nidhi Gaur. So Dr. Nidhi Gaur earned her PhD in organic chemistry from IIT Kanpur in 2010 under the guidance of Sandeep Verma. So winning second prize in Lilly Asia Outstanding Thesis Award. She completed her postdoctoral research at TIFR Mumbai, Albert Einstein College of Medicine in USA, the University of Geneva, Switzerland, and the CSGI, University of Florence. She is currently an associate professor at International University. Her scientific contribution is her research is on molecular self-assemblies, spans biology and material sciences with publications in top QN journals like Current Opinion in Chemical Biology, ACS Chemical Neuroscience, Soft Matter, and Chem Biochem, cited in Nature Nanotechnology and ACS Nano. A NASI member, she serves as on the SERB Sure Screening Committee, reviews for ACS, RSC, Elsevier, Springer, and referees for UGC FRG proposals. She collaborates with Professor Hud Gazit, Tel Aviv University, and has organized the DST Inspire Science Camp, Guj Cost webinars, and international e-conferences. Today's her talk is focusing on investigating the role of metabolite assemblies in the etiology of inborn errors of metabolism. With this short introduction, I would like to welcome Dr. Nidhi Gaur.
The very nice introduction, and thank you to the SID organizing committee for giving me this opportunity to present the research work of our group. And so the title. So I will be today speaking about one of the very interesting research area which is going on in my lab, and it is on the implications of self-assembly in the pathophysiology of diseases. And the title of my talk today is on investigating the role of metabolite assemblies in the etiology of inborn errors of metabolism. So before proceeding, I just want to make people familiar with self-assembly, which most of you might be knowing. And this is the spontaneous process of self-organization of molecules to well-ordered structures, which is mediated by non-covalent forces of interactions like hydrogen bonding, pi stacking, etc. And the molecules assume to form the most well-ordered structures, which are energetically favorable. So these structures have wide implications in material science and biology, and they occur in nature like cell membrane, DNA. They are all formed by the self-assembly. And so coming to metabolite assemblies, the process of self-assembly is not always favorable. And it's in the previous reports, it has been shown that in the Alzheimer's and Parkinson's disease, some proteinaceous metabolites, they accumulated and they are referred to as amyloid. So conventionally, it was considered that amyloids are only formed in the brain of Alzheimer's and Parkinson's patient, but recently it was shown that even non-proteinaceous metabolites like single amino acids, nucleobases, glucocerebrosides, they form amyloid-like structures, and they form amyloid structures because the enzyme which is responsible for the catabolism of these metabolites, they get dysfunctional, due to which such toxic structures are formed, which induces cell death. So there are many diseases like phenylketonuria, maple syrup urine disease, or tyrosinemia, to name a few, which are formed by the excess of phenylalanine, branch chain amino acids, and tyrosine. Uh, and the first groundbreaking report was given by Professor Ehud Gazet in this context, that a single amino acid phenylalanine, it forms amyloid-like toxic structures and disrupts the cell. And he further extended this hypothesis to other metabolites like adenine, orotic acid, and they found that they also bind amyloid-specific dyes, and these structures are indeed amyloid in nature. So there were many subsequent reports which showed how these structures are amyloidogenic, how uh, they induce cytotoxicity, how they create a lethal tap, they disrupt cell membrane, and how they induce apoptosis. So coming to our work, uh, we for the very first time showed that non-aromatic amino acids like cysteine and methionine do assemble to form amyloid-like structures. And I'm saying this for the very first time because earlier it was known that only aromatic amino acids have this propensity, but our group for the very first time showed that cysteine and methionine non-aromatic amino acids also assemble to form amyloid-like structures. And in this paper, we showed that cysteine, it assembles to form like this fractal fibril-like structures, and methionine, it assembles to form this long fiber structures, and these structures were characterized by SAM optical microscopy as they were big enough, and then by transmission electron microscopy we showed that these structures are soft, and by XRD analysis, by this solid state NMR, FTIR, TGA, we showed the importance of hydrogen bonding in causing these self assemblies, and then we collaborated with our collaborators and found that by computational simulation, by MD simulation, that there was some hydrogen bonding interactions which were leading to formation of some aggregates, and solvent dependent studies and urea studies, we further uh, substantiated this hypothesis that hydrogen bonding is the main factor which causes this aggregation. And then we did amyloid specific dye staining like congoid and thioflavin T. They only bind to amyloid structures, and we got positive results. And further, we did MTT assay and we showed that these structures were cytotoxic. So in continuation with that, another ACS CAM neuroscience paper we published where we showed unusual aggregates formed by the self-assembly of proline, hydroxyproline, and lysine. And in case of proline and hydroxyproline, there was concentration-dependent amyloid structure formation, while in lysine, it was age-dependent or it was time-dependent amyloid structure formation as assessed by AFM and other microscopy. And we have carried forward, we are carrying out, we are studying different branch chain amino acids, and in case of isoleucine, leucine, valine, also we have characterized such amyloidogenic structure formation using same biophysical techniques. Then here we also did some cross-seeding experiments with amyloids and urea, and also the MTT assays on the test showed that these are significantly toxic structures. This is another work which was published in ACS Chemical Neuroscience recently, where we showed glutamic acid and aspartic acid assembly to amyloidogenic structures, and how these structures stain the dye, how by various MD simulation studies, what are the forces which are responsible for formation by solution state NMR, how hydrogen bonding is responsible. Then this is another thing. So far I have showed our research on single amino acids, but we extended this to other metabolites and other metabolites of uric acid pathway, urea cycle, etc., which is again published in ACS Chemical Neuroscience. And here we see this metabolites ornithine, citrulline, and showed that these ornithine and citrulline metabolites of urea cycle and this hypoxanthine and xanthine of uric acid cycle, they assemble to form very peculiar structures, and these structures are cytotoxic and stain uh, with the thioflavin T dye. And in this study, we also did in vivo cytotoxicities assay on C. elegans. And we did, along with behavioral assays, we did some HSP assay, shock pathway assays, which showed that those C. elegans were treated, which were treated with amyloidogenic structures. During the HSP assay, they showed very, like very, they showed very rapid cell death, and there was very, the cell death was much more. And HSP as a shock protein, this assay is also very important because typical amyloid structures, they affect the HSP pathway, and then they produce cytotoxic behavior. So this again shows that it is a characteristic of archetypal amyloids, which are conventionally studied, and other metabolites from other pathway also, if deleted, they can form this. And the other very interesting thing about these study was that these soft structures gradually change to crystalline structures. And in the diseases are caused by the accumulation of these metabolites. These are Leishman syndrome or gout, where the crystals get accumulated at the final stage. So this study shows that first the amyloid structures are forming and then they are transforming to crystals. So then we extended this to other metabolites like homogentisic acid, N-acetyl aspartic acid, and isovaleric acid published in Chem Biochem. And here, phenylketonuria is caused by accumulation of homogentisic acid. Canavan disease is caused by accumulation of N-acetyl aspartic acid and isovaleric acid. Acidemia is caused by the accumulation of uh, valeric acid, which again we did the same studies, similar studies, and we did many biophysical assays, and then this many assays on C. elegans model, uh, flashing assay, pharyngeal pumping assay, shock assay, and we concluded how these cells, how these C. elegans are getting affected, how the mortality is getting affected by these amyloid structures. So all this works we have done in collaboration with CDI Kanpur, and then uh, we are recently studying amyloid-like aggregation properties of nucleobases on C. elegans model, and in this context we are in this stage of microscopy of this cytotoxicity, and this paper is almost ready. So conclusions were from these studies were that the metabolites of non-aromatic single amino acids also do have propensity to form amyloid-like structures. From the metabolites of urea and uric acid pathway, we got an important observation that the amyloid-like fibers will change to crystals, and then they may cause these diseases, gout and Leishman. And the in vivo cytotoxicities produced in C. elegans model also confirm the cytotoxic nature of the assembly, and the e-shock assay suggests that they may affect HSB pathway, largely typical of the Alzheimer's amyloid. And in future, we want to study it by cross-seeding and C. elegans Alzheimer model. What will be the effect? And the other metabolites like homogentisic acid, etc., they also assemble, and the nucleobases also assemble. So what's next is we have a very good collaboration with Blavatnik Center of Drug Discovery at Tel Aviv University. So we have shown these results, and ultimately we want to talk about common arrangement of these amyloids and the amyloids. We have to compare the amyloids formed by Alzheimer's and Parkinson's disease, and we have to assess like if the Alzheimer's, the common FDA approved drugs of Alzheimer's or can be used for treating these rare metabolic disorders also. So this was just one research area. There are other research area which is going on in my lab, which I've just shown in one slide, like modified single amino acid assembly, where we take very small modified single amino acids and we assess their assembly and we show their material science and biological applications. Currently, we are studying their and like how they can be used against the antibiotic resistance. Then this is another very interesting area, which is on heterocyclic compound self-assembly, where we assess the self-assembly of heterocyclic compounds and assess like what is their implications of self-assembly in the photophysical properties. Uh, and on the sensing, research has been appreciated by the community, and my students have regularly received best poster and oral presentation award in prestigious conferences like JNOST and International Conference. And this is acknowledgement to Vice Chancellor Professor DJ Shah, Research Director JS Yadav, all my PhD and postdoctoral students, without whom this work is not possible, and all my very kind collaborators who are always ready to help and provide their facilities, and most importantly to the funding facilities, the funding served, and in Rashid University for the infrastructure. And thank you so much to all for this.
Thank you, Dr. Nidhi, for finishing your talk in time. So I'd like to listen for question and answer. Any questions? Okay. Yeah. Thank you. So
So,
very nice presentation, and you have fantastic publications of your own.
yeah,
Yes, Chem Neuroscience is
yeah.
a prestigious journal. Always this thing. Okay, with this, uh, after this, I will hand.
Thank you so much, ma'am. I request Dr. Jankiram Vetla to felicitate Dr. Niligor.
Good morning, everyone. Next speaker of this session is Dr. T. Pavan Kumar. Dr. T. Pavan Kumar is dedicated researcher with expertise spanning organocatalysis, asymmetric synthesis, nucleic acid chemistry, process chemistry, and medicinal chemistry, natural product synthesis, and flow chemistry. His work integrate fundamental research with practical application, particularly in process and technology development, where he specializes in optimizing synthetic methodology and scaling up the chemical process. In addition to his research, Dr. Kumar is actively involved in intellectual property management and business development with proficiency in patent research, market structure analysis, patent drafting, and portfolio management. He also has a strong academic presence, teaching courses in organic chemistry and intellectual property, including patent search and analytics. His experience extends to technology development, process demonstration, project execution, client management, and comprehensive documentation, making him a valuable contributor to both academia and industry. Title of the talk, Patenting Chemical Innovation, Science versus Scientific Art, this synergy. Now, sir.
So
Stage is yours.
thank you very much, and I really thank Indra University and also the team SIDD 2025 for taking all the efforts in inviting me to this program. I'm very much thankful to Dr. Yadav, who has been instrumental for nurturing people like us, and also my special thanks to Dr. Vishnu Maya Bisai, who could take all the care to invite me here. So today I'll be talking to you on patenting science versus scientific art, the synergy. From last 2 days, you are listening about the science part of the activity, but now we'll see how science can be integrated with art so that we can achieve some productivity. And preferably, I'll make this session not as a lecture, but rather it would be an interaction. So these aspects I'll be covering. Some slides will be there where only you need to see so that you can understand. There'll be few slides where we need to interact or discuss, and there'll be few slides where the queries. I expect your participation with the relevant answers. So I will be covering all these things. Now this is the season of 2020, but I have to play only 20, not the 2020. Okay. And innovation. The title of this conference has got a word innovation. When we talk about innovation, people define innovation in different ways. Believe me, the impact of innovation is it is only through innovation we can distinguish between a leader and the follower. If you wish to become a leader, definitely you should innovate to whichever field you belong to. And there were several definitions on what is called invention and innovation. But believe me, a simple definition is if your idea leads to a first solution, that is invention. And if the same idea after becoming solution brings you profit, that is innovation. And for that, you need to come up with a smart idea. Ideas which are smart only will survive. Others will not survive in any case. Today, whenever you have an idea, whether you are a researcher or working at industry or an academician, everyone develops ideas. But our major practice is these two. But now the requirement is this, demand is this. So then how to tackle with this? Irrespective of whether we belong to industry or academics, we are in this phase of transition. And whenever you are in whenever you are in such a phase of transition, always you should take an IP first. Take an IP, you will also get the publication for the same work to whichever journal you submit. It is like 1+1 offer provided if you take IP first. Reverse is not possible. And it's a high time. If we innovate, people will still recognize us. Otherwise, we'll be ignored. Ignorance is equivalent to that. So therefore, the situation is like the times of 1942, where the slogan was "Do or die." And this would be a scenario when you do not have IP asset. Big will eat small, small will eat further small. Being small without IP is so dangerous. And if you have IP, whatever small you may be, you are very strong. And here, for sake of students, I made this slide. This is the classification of intellectual property. Of course, I don't go into the details of that, but then whenever you come up with an idea, there is a possibility to obtain any kind of IP depending on the kind of the product that you develop. We are talking about patents, and they are an outcome of an invention or innovation. If you come up with an invention or innovation, definitely you would get patents. And IP is a property. It is a limited time enjoyment property. You cannot enjoy for long. If you have a bungalow or if you have a car, if for generations together you can keep it. But IP is not like that. Specifically, patents, they are enjoyable for only 20 years, provided if they are granted. And then why IP is required? Because anyway our days are going, then why today we suddenly started talking about patents? When we talk about scientific contributions with respect to publications, we are number 3 in the world. Very good, we should celebrate, but we should not stop there. When we talk about patents, China contributes to more than 47% of the patents worldwide. And look at EPO, Japan, USA, and Korea. Rest of the world contributes to 15%, where India also belongs to. India is, along with more than 190 countries, contributing to about 15%. And if you ask me the specific numbers, about 10 years back, Chinese patent office used to receive 6 lakhs patents, but now it is receiving around 15 lakhs. Indian patent office around 45,000, now it is about 60,000. So there is slight increment, there is a slight increase. In last 2 years, see 15 lakhs last year, this year 16 lakhs from China, but India 55,000 to 64,000. And these are the top 5 countries. Then what is called a patent? We keep coming across this term very frequently, but if you read the definition from several sources, they'll go in several lines, maybe a paragraph. But in simple terms, a patent can be defined as a contract between the government and the inventor. It is as simple as this, for which government gives the inventor the right to exclude others. Once you get a patent, you have the right to exclude others. In simple terms, we can refer the patent as a techno-legal document. Technically, we all are very strong. Whatever we were discussing for last 2 days, it was all about technical part. Today, I'll show you how this technical part can be integrated with the legal component so that we can bring some patents. I have to go this way. It is not that protection of inventions is happening very recently. It dates back to 500 BC, where Greeks used to grant protection to the inventors. This is a very, very old practice. Maybe the terms or the kind of terminology or the words we are using are new. This is the first patent from US in the year 1790 for the process of making potash, again a chemical process. And why America is so powerful? IP, their strong IP is also one of the reason. I think many of you who might have visited abroad, you would have witnessed it. If you enter into the American Patent Office, it says like this: the patent system added the fuel of interest to the fire of genius. Abraham Lincoln himself has got a patent in the year 1849. And this is the scenario. Look at how America is the first country to come up with a very, very systematic practice of patents. By looking at US, Japan started, it also became very much advanced. Then started Europe and Korea. Chinese first patent has come only in the year 1985. Before 1985, there was no patent from China, but now it is the world leader. And even Prime Minister in one of the recent science congresses mentioned about innovate, patent, produce, and prosper. These 4 steps will lead our country towards faster development. Then what is a patent again? Patent confers the right to exclude, but not the right to do. So therefore, it is referred as a negative right. And that means, as an inventor, if you get a patent, you have the right to put a red signal. orange or green, it is up to you, but patent law do not say anything on that. One product can have one or more than one technologies, whereas one technology can involve one or more than one patents. But believe me, if you have one patent for one invention, if you have more than one invention, you have to file different patents for each invention. And let us take one quick query, majorly for the students. W invents and patents the wheel. B later invents and patents bicycle. The query is this: who can make and sell the bicycles? Can anyone give quick answer, please? It is 2020 match. Yes. Maybe bicycle. There is W and B. You mean B? Okay. Any other one more response, please? Quick response. Both has to combine. Okay. Yes. Yes. Okay. Then anyone? Yes. Yes. It's B. Okay. The answer is here. Neither W nor B. If it is W, they can sell only the wheels. They have to do like this only. Okay. And if it is B, B can make wheelless bicycles. You have to ride bicycle like this. So therefore, this is the importance and impact of it. It is not that all the time whatever you develop and whatever work you get patent, you have to practice. No, make a patent, sit at home. Even then you can attract money, like Coca-Cola ring pull system. So therefore, we should understand more about patents in detail. If you feel that your work is very innovative, very novel, it doesn't mean it will definitely attract patents. It should satisfy the patentability criteria. In short, it can be referred as NUNS test, NUNS test: novelty, utility, non-obviousness, and statutes of the nation. First three are quite common in all countries, but the fourth one is, it is also common, but it differs from country to country. I don't know how many of you have seen the patents. Majority of the faculty might have already seen, but for the students, irrespective of the number of pages goes into the patent document, there are two major parts. One is the disclosure, which is useful for the public. The other one is claim. As an inventor, for you, it is the claim which is more important. If your claims are strong, you are strong. And a document is called as a patent, provided if it has got patent claims. There should be at least one claim. Without claim, it is not a patent. Now, another query. If you are making fence to your property, which fencing do you prefer? Faculties can also become students, irrespective of the place from where you belong. Which fence? You cannot take this much time. If you take this much time, gone. If we have to be in the race, it should be spontaneous. First, fourth, others don't want fence. You want to make your property open. Huh? Combination of 3 and 4. Some are close to the answer. Many are not close to the answer. The answer is only 4. Whoever says only 4, they are in the direction. Because it is the claim which defines your invention. So invention is always defined by the claims. Claims are tested before granting by the examiner. So whatever you put in the patent document, they don't see the entire document. They only look at the claims. If they are proper, your patent is strong. If they are not proper, you don't have any chance to fight with them or discuss with them or submit any of your request. Because disclosure supports the claim, but it is through claim you get the right. So therefore, claim is regarded as heart of a patent. And that is where the art lies. It is an art based on the science that we practice. What are the different types of claims that generally we come across? If you develop a product claim. If you develop a process claim. Even product come process claim is also possible. Marcus claims, especially for the medicinal chemistry and organic chemistry people, we have a great advantage of making Marcus claims. A generic claim which can protect a large number of specific molecules. Make 20 molecule library, but once you make a Marcus claim, any kind of molecule related to that generic scaffold cannot be done by others. So it protects everything. I'll show some good number of examples from our own research. Then Jepson claims, improve upon the existing invention, you will get a Jepson claim. And Swiss claims, second and further application claims are called as Swiss claims. Generally, they are referred to the medical use, and composition claims are also there. So these are the different kinds of claims that generally we come across. Fine, but it is not that all the claims are allowed in India. Every country has got its own patents law and their practices. In India, only these are allowed. Product claims are allowed, process claims are allowed, composition claims are allowed, method of treatment not allowed, new physical forms generally not allowed unless and otherwise if there is some enhanced therapeutic efficacy. But believe me, as such, there is no definition on what is called enhanced therapeutic efficacy. Neither Supreme Court gave the definition, neither nor patent office has the definition. And there's a famous case study of imatinib. And living organisms are not patentable except for the microorganisms. Few examples I'll show. There is a patent for cortisone from in the year 1940, and here is the patent number. And then a patent for antibiotic in the year 1949. And during COVID times, though the entire world has suffered, patenting activity did not stop. There were at least more than 5,000 patents which were filed in the span of 1.5 year. 2020 January February से लेकर 21 June तक more than 5,000 patents worldwide. If you ask me what are the numbers from India, at least there are about 172 patent applications filed from India during that time. That means, and after having some experience in patents, we thought on one fine day we came across these lines by Albert Einstein. If you always do what you always did, you will always get what you always got. Then we thought, how can we orient towards this? We were discussing after several weeks. Anyway, I was having some knowledge on patents. Why can't we integrate this knowledge in our research? So that is where we started practicing IPT. This is what we were doing. I was doing as a PhD scholar or as a DST Inspire faculty. But then gaining some experience on patents, then we thought we should try to integrate our knowledge on patents with the research. And by now we could file more than 46 applications, out of which are more than 25 are already granted in several countries. Luckily, 4 of our initial patents, we could license them to Sun Pharma and we could attract 5 crore royalty. So with this story, I am here to 2 minutes. Okay. Science and scientific art. This is science. We developed our 9 compounds here, but this is how we made the market structure. And another one on indole sulfonyl benzamide derivatives. Science is made a library of 30 compounds, but this is the market structure. So now no one can make any kind of analog with this structure. And see, these are the different countries where we filed. First one in 2 countries, but the second one, indole sulfonyl derivative, we filed in more than 8 or 9 countries because that is very much selective towards HDOC inhibition. We have 2 molecules which are very selective towards HDOC6 and HDOC8 inhibition. And these are the patents. And there are few more works, science versus scientific art I just mentioned. And there is a process patent for zafirlic acid. It is an approved drug molecule for asthma treatment. And as on today, I believe this is one of the shortest process that we developed, for which we filed patents in both India as well as US. Both are granted. And there is also a patent for spiroxindole compounds as GSK3B inhibitors. These 2 we filed in India and US. These are the front pages of the patents. And then another work, look at this structure. When you look at this, you don't believe that this would be an organic structure or chemical. But yes, these are the compounds that we made in collaboration with IACB Kolkata. We filed the patent in India. And another one is a new process for lipoic acid. I just talked to you on Japan claims. We have repeated the process as such, whatever is already known, but there is a slight improvement. That slight improvement is a very, very significant improvement. We only changed solvent system. All the reported methods are having either 2 or more than 2 solvent systems. We developed the process only 1 solvent. There were minimum 3 purification stages, but we developed that with 1 purification. It is an approved drug molecule for the treatment of a kind of diabetes. 96% import is happening from China, 4% from US. Even as on today, no Indian company is making this. Biocon gets it from China, then makes final stage purification and sells in the market. We were trying very hard to commercialize this, but due to some reasons, it is getting delayed. So these are the patents for those two works. And I had shown you about American patent and European and WO patents, but now you see Indian patent. The front page of an Indian patent looks like this. It is not that organized because our patent practices are very, very immature. And this is the very first attempt where we thought of filing patent. I was just patent got licensed. Simple thing, what we did is SK1, then we tested. Because of the biological activity studies, we could file patents, and because of that, we could attract royalty. And this is another. This is the first patent. The second one is on the process of making nicotine. Three-step process. Both it can be done stepwise as well as single one pot process. There are the patents for that, and then there's a front page of the patents. And believe me, it is not that all the time you need to come up with 100% new work. 90% of the patents worldwide are granted for improvements, and improvement is nothing but innovation. And there's a great drawback in our practices. People say, I am filing patents, I am writing the manuscript for a patent. Whenever you use these kind of words, the other one, if they have some knowledge on patents, they will definitely catch you. While practicing patents, these are the terminologies that you should use: inventor, specification, drafting, and assignee. And future wars will not be fought over territories, but definitely over exploitation of ideas. So therefore, research orientation has been majorly towards making impact factor publications first, but now the time has come. Along with that, we should also reorient towards making India first, innovation first, and IP first. So IP has to be taken care. For this, we are doing several programs. These are some programs, and I think this is the last slide. And I thank Dr. J.S. Yadav. This is the first photo when I met him about 12 years back. And then, thank you. If you have any queries, we can take one or two.
Now session is open for question answer.
Yes, yes.
We publish it, and later we realize that okay, can we file patent? So what is the rule that once you publish that, can you file the patent application in India?
People say within 1 year of your online publication, still there is a possibility of filing patent, but it is possible only in the countries like US, not in India. In India, also possible, provided if your publication happens in a government gazetted approved program. Suppose in this conference, if you present here tomorrow, if you wish to file a patent, not possible.
Okay, thank you.
So therefore, please make patent first, then go for publication. And you need not wait until it gets granted. File it, then publish your work.
Okay, thank you.
So after filing patent, I was asked whether technology is licensed or not. What is technology license
See,
mean?
technology licensing and having patent are two different aspects. You can license technology after having the patent. You can also license technology without doing any patent.
So what is licensing?
Technology license, you developed a technology, but it is not scaled up to a higher level. If some industry or some other party is interested, you can transfer your know-how or the process to them.
Oh.
For that, you will attract some money. You can fix a price so that it can be transferred to others through a licensing agreement. There are again two types again: exclusive or non-exclusive. Exclusive means only you will be licensing to them, and you will not be able. You should not license to others. Otherwise, it can be licensed to me.
So between two parties, like the
Yes.
Okay, thank you.
Majorly two parties. Yes.
Can I transfer to the several companies the same as a technology?
As a technology, if you talk in terms of technology, yes. As on the basis of non-exclusive licensing agreement, you can transfer to several people. But if you transfer it as a patent, generally it is not possible because majorly companies ask to assign your patent to them so that they can limit others from practicing.
Yes, sir. I request Dr. Kyari Rana to felicitate Dr. Pavan Kumar. I will request Dr. Pavan Kumar to hold on the stage. And Dr. Pavan Kumar has been very kind to bring a gift from IMMT to Industrial University. I will request Dr. J.S. Yadav sir to kindly come on the stage and receive the gift.
And also I request kindly clap.
Yes.
Sir, it is a token of affection.
Appreciation.
But token of affection. From IMMT. The Polar Chakra. So whoever visits Odisha, this is correct Margaret.
I sincerely thank Dr. Pavan Kumar.
Yes. Different designs or different order size, different size, different design.
Now, next speaker is for this session, Professor Ramendra Pratap. Professor Ramendra Pratap joined the Department of Chemistry in 2010 and became a full professor in 2022. He earned his PhD from CDRI Lucknow under Dr. Vishnu Ram and pursued postdoctoral research at the City University of New York. He was awarded the Alexander von Humboldt Fellowship to work at University des Saarlandes, Germany, 2009 to 2010, with Professor Oli Kasmer. With 150 plus publication in international journal, he has authored 2 books on heterocyclic chemistry, 2 reference book on pyrene, and many more book chapters. His research on carbocycles, heterocycles, metal catalyzed bond formation, nucleoside modification, and medicinal chemistry, securing funding from DST, SCRB, CSIR, ICMR, and others. He has been a visiting professor at Kyoto University, Japan, GSPS Innovation Fellowship at Visitor Germany under the AVH Alumni Program. He also served as consultant for Frontier Course South Korea, aiding in the development of biological active molecules. And it's a short introduction. Now, title of the talk: Cyanomethylarene develop new molecular entity. Now, sir, I would like to invite on this stage. Thank you, sir.
Before going to start, I say that Dr. Sally was my student.
Mic not on.
Mic is not on.
Mic is not on. Mic.
So as I was mentioning that it's always great that your student introduce you at some forum, and I'm so much happy that she is doing good here also. And before going to start, I would like to thank the organizer, Professor J.S. Yadav, Dr. Vishnu Maya, Dr. Sally, and others for inviting me for this conference. And today I'm going to talk about cyanamethylarines as a source to develop new molecular entities, because usually what I believe that any research, if you start with something, you can find that one molecule can be used as a source to develop enormous number of molecules. And I will give you a few examples out of which you can see that precursor has been taken, and using that there are researchers who created almost all kind of the heterocycles, carbocycles you can imagine. they have been able to develop that molecule just by starting with one of the precursor. So here today I am going to just discuss that how cyanomethylarenes can be used to develop a large number of or large class of molecules. So now here there is some reason which makes me to take this molecule as a starting material, and out of that the point one, that the stability, because if your starting material is not stable, maybe in some of the reaction condition it decompose before it gets get reacted. So it's always important that the stability of the starting material should be taken care. Then what kind of property it can have? So you can see that this carbon, this can give the protons and it can generate the nucleophile during the course of reaction, and nucleophile mediated reaction is one of the class of reaction which has been enormously used, I must say, that in each and every lab. Then the cyanide group, it can act as the electrophilic center also. That means you can involve the nucleophile intramolecular or intermolecular to generate the new type of the functional group within the molecule. Then if you are going to incorporate some orthophilite group, that functional group has also possibility that they can involve in the reaction and they can go for the consecutive cyclization or replacement to give you the new kind of the product. And definitely this is interesting molecule for further exploration. And due to less exploration, we thought, okay, let's explore this molecule for further chemistry. So ketene diethyl, Professor Ila and Junjappa, I think he worked a lot. Even one Chinese group, the Qinlu, he was working on this molecule. And this ketene diethyl has been used to generate, in my knowledge, you can imagine so this molecule can be easily synthesized just by reacting with carbon sulfide and methyl iodide at low temperature with sodium hydride and THF. It is 1 hour reaction, and you can get this ketene diethyl. So as I said, here what we have done is we have taken ortho cyanide group and then the cyanide also. So that means here you have two type of the cyanide group, one which is in the benzene ring and other it's here. So this cyanide group is playing role to making this double bond just for the source of the Michael addition. And then further we thought, okay, let's utilize this molecule or this group for further cyclization. So using this molecule, we perform 1,5 annulation reaction. And very simply, when you are using the different type of reaction condition in KH and DMSO at room temperature, in 2 hours, we are able to generate good yield of this compound. Yes. So after optimization, we just synthesize different type of the product over here, and you can see that we can synthesize dinaphthyl ketone, then different type of aryl naphthyl ketone, having this ortho amino. So usually the putting amino group at ortho position to the carbonyl group. This is also precursor for new type of the heterocycle. Maybe you can go for the benzoquinolines and even the indole kind of nucleus can be created over here. So this molecule do have the importance. But in the same reaction, when we are using this halide substituted substrate, so in addition to the usual product, there was replacement of this halide with the S-methyl is also observed, because during the Michael addition, when the S-methyl is coming out, this S-methyl was dissolved in the solution and that attacks over the via ipso attack at C4 position, because that is in conjugation with this electron withdrawing group, and that's how the ipso attack followed by loss of halide was giving this compound also. And you can see that the major compound, that means with halide, was this one, and the minor amount of this S-methyl containing compound was also observed. So to further explore the reaction, because if you have to perform the amination reaction in aromatic system, you have to follow the catalytic approach. We thought, okay, just do the simple nucleophilic substitution over here, and then we use this as a precursor. So maybe we can directly introduce amine group in the aromatic ring, and finally we succeeded in this also. So this is the simple mechanism for this 1,5 annulation reaction, and you can see that under basic conditions we can generate the inulate, and this inulate can add over this double bond via Michael addition followed by loss of methane thiol to give this intermediate, And in presence of excess of base, this base can again abstract proton over here, which can cyclize with this nitrile group to give this product, and this can enolize to give this final product. So this the purpose to show this other pathway, because this reaction is also feasible, where there was possibility that out of these two cyano, the reactivity can be observed with this also. And you can see that if the reactivity will go with this cyano group, then there is possibility to generate this pyran group. And later on, I will show that if there is no cyano group present on this ring at ortho position, definitely you can go for the preparation of this pyran ring also. So this is the structure confirmed by single crystal X-ray. So since we are synthesizing by 1,5 annulation approach, we thought, okay, let's go vice versa or other way around. Then we perform the retrosynthetic pathway. So this is the pathway I just discussed. Now I thought, okay, now we can just take this ketene diethiacetal and we directly use this as a source of nucleophile to see whether 4 plus 2 cycloaddition can also give the same product. And interestingly, this reaction was much more better than the other one. So in this case, in earlier case, the yield was like 60-70%, but in this case, you can see that under the similar condition, we are getting 94% yield. That means changing the substrate is just changing the yield of the reaction. Yes. So at this reaction, we also went up to the gram scale, and there was pretty good yield we have observed in this case. And even in this case, we have also used the aliphatic substituted ketene diethiacetal, and in those cases also we got the good yield. So since we are doing this reaction, so as I said that this S-methyl group was replaced. This is a simple mechanistic investigation for that. And what we propose that due to ipso attack of this already present methyl group inside the reaction mixture, you have this intermediate which was stabilized by this carbonyl group. And then when this reversal of negative charge is observed, halide can go out and we can observe this product B. So since this substitution reaction was observed, we thought, okay, let's take ortho substituted group and maybe we can utilize this amino group generated in C2 and we can cyclize to get the new molecule, and definitely it worked. So when we are performing the same reaction with ortho substituted halide, you can see that in addition to this S-methyl group, we were also able to synthesize this benzoacridine molecule, and depending on the how long we are running the reaction. So for example, if you run the reaction for a certain time, we get 3 products. But as soon as we run this reaction at higher temperature, then compound 1 with halide is completely diminished and we just have this compound B and C. That means that S-methyl group cannot be replaced with this nitrogen nucleophile, but halide can be replaced with the nucleophile to give the acridine. So just to confirm this, we have directly taken this halide substituted naphthalene and we just perform this reaction at 100 degree centigrade, and in 30-40 minutes we are able to completely convert this molecule to benzoacridine in quite good yield. So this is the mechanism again in this case. So earlier we have seen that the histophenone was nucleophile, but here there is nucleophile source is this cyanomethylarene is itself. So after generation of this cyanomethylarene nucleophile, there is Michael addition followed by loss of this methane thiol. We can get this intermediate, and this intermediate can have possibility of these two isomeric structure, that is E isomer and Z isomer. So E isomer has no possibility to interact with this orthonitrile group, but Z isomer has this reactivity with orthonitrile group, which has possibility to give this product. And if there is possibility of this isomer, then there is possibility of generation of pyran was also there, but formation of this product confirm that probably there is formation of Z rather than E isomer. So further we explore this reaction, and using this reaction, now we have used this ketene diethyester and nitromethane as a nucleophile source. So you can see that we can generate the naphthalene with the nitroaminonaphthalene, which can be directly converted to diaminonaphthalene. And you know that the diamine compound, they are very versatile to synthesize different type of heterocycle which can give the different type of biological activities. So this has been explored to synthesize different type of the heterocycles also in quite good yield. And these molecules they were already tested also, and they do have the very good result also. Yes. So as I said, single starting material, further exploration. So further we explore another nucleophile source with this synthone, and you can see that we are able to synthesize the benzocarbazole. We are able to synthesize this arylated naphthalene. So earlier you are synthesizing this COPH. Now directly aryl group can be inserted in the naphthalene group, and here we are trying to have, so when we are taking this, our thinking was that we may synthesize this benzophenethylene molecule also, but somehow we could fail to this. But yes, these two reactions we have got in moderate yield, and these compounds also allows to reduce many steps for CC bond formation, which we have to require later on. Then these are some other reaction. If you use the ammonia source like urea, we can synthesize isoquinoline, and this is the mechanism. So in this case, if this S-methyl is replaced with NH2, then this can cyclize with this orthonitrile group to keep this compound. And hydrogen was also used to synthesize this compound. So if you are using this hydrogen as a nucleophile source, you can see that we can synthesize pyrazole fused isoquinoline molecule in quite good yield. So same starting material. Now we thought, okay, let's think about some other nucleophile source, and here we have used the ketene diaminerals, because in this aminals there was possibility that either this NH directly act as a nucleophile source, or it can enolize and this carbon can also act as a nucleophile. But in this case, interestingly, we are able to utilize this nitrogen at first, and later on this, rather than involvement of this carbon and this cyanide, there was enolate, or you can say this O minus further reacted this, and we are able to synthesize this oxygen molecule. But actually what we were thinking, that maybe this further resonance can give this enolate, and this carbon can cyclize with this, which we could not able to get it. That means here the selectivity went in other direction. This is another very interesting reaction, and here we have changed the starting material from ketene diethiacetyl to this 2H pyran 2-one. And in this case, when we were performing the reaction, there was consecutive cyclization reaction, and we were ending up to tricyclic compound, that is the benzoquinoline, with the too many functionality, that is nitrile, arylation, and diamino compound in this case. And in the beginning, what we were thinking, that we may get phenethidine molecule, which I will show later on, but somehow we are not able to get the phenethidine, and we have ended up to this very nice benzoquinoline molecule, which was confirmed by single crystal X-ray 2. So this was the optimization reaction, and sodamide was the base which was acting well to give this compound, and we got 82% of yield of this compound. And this reaction was carried out in both microwave as well as the usual reaction condition. And the thing is, you can see that this reaction required longer time, and up to 35 hours we have to run this reaction to get the product, and that's why we thought, okay, let's move to the microwave. And microwave definitely, in my belief, 50 minutes is still longer time, but after 50 minutes we are able to convert this compound in microwave to this benzoquinoline molecule. So these are the different compound what we have synthesized in this case, and these are the microwave as well as we have utilized this one. So mostly the yield in microwave somewhat better, but traditional heating was also giving us quite good yield. So this is the mechanism, and what we were thinking, if you can see that in our thinking we should get this molecule, but the reaction gave this one. So what we were expecting, that due to extended conjugation of this double bond and this R group, which is aromatic, that also stabilize the negative charge over here. So we thought this negative charge will attack over via 1,6 Michael addition over here to give this intermediate. And after this intermediate formation, there is a possibility, and this can decarboxylate. And after decarboxylation, there was two pathway what we proposed. Pathway one was this one, where there was possibility that in presence of excess of base, this hydrogen can be abstracted. This minus charge can conjugated with this double bond, and this can attack with the cyanide, and that can give you this imine, which can conjugatively cyclize to give this phenanthridine molecule. So that was our expectation. But here, I think due to the steric region, rather than reaction going in this pathway, here the allylic attack of this nucleophile is observed with this nitrile group, and this imine further cyclized with this nitrile group to give this final benzo-C-phenanthrin molecule. So as I say, the same compound in different reaction condition with different substrate, you can have different type of the product. And this is one of the example. So you can see that when we are performing this reaction with simple non-fused 2H pyron 2O, there was formation of this molecule. When there was fusion, then there was formation of benzoyindene. And there is justification for that, but we were thinking that we may be able to make some complex molecule using this chemistry. However, this reaction is quite good. However, the yield was not so good, but still we are able to synthesize this compound. So you can see that in KVH DMF 100 degree centigrade in 2 hours, we were able to push this reaction up to 42% yield. And after getting this, we synthesize different type of the compound. So what we found that we are also able to synthesize this intermediate. And this intermediate, we further use this intermediate to synthesize this compound, and we found that there was further oxidation reaction is observed, which are giving this compound in the reaction. Okay, so now there was the structural comparison what we have done, and we found that there is no substituent in this case. If there is no substituent, the benzoyl formation was observed. But if there is substitution, then the reactivity varies. And here you can see from the mechanism that following the same pathway after decarboxylation, in this case we end up to this. And if you go for the same cyclization what we observed earlier, there is the formation of very sterically rigid molecule, and that was not supported in this case. And therefore, since there was no possibility of this reactivity, so in presence of the molecular oxygen in the solvent, we propose that probably there is further oxidative cyclization is observed, and here there is a attack of this nucleophile at the oxygen, which can give this peroxide bond. And further this nitrogen on amine cyclize with this with the loss of hydrogen peroxide to give this molecule, and in presence of excess of base, this proton can be abstracted and this can neutralize to give this final benzoyindene found. So both the structure has been confirmed from the single crystal X-ray, and also this has been confirmed that this is the intermediate of this reaction. And further, in the beginning I have mentioned that there was possibility to incorporate this cyanide in the reaction, and this is the proof of that reaction. So for example, if there is no ortho group, we can synthesize this compound in quite good yield. So this is another reaction, and this is oxidative decyanation, and after decarboxylation we can decyanation and oxidize this molecule, and this reaction was also quite good. So you can see that here after this benzyl cyanide group can be decyanated and to give this carbonyl group over here. So this is the last one. Since when we are using this, you can see that sometimes it gives oxidative decyanation, sometimes it gives the oxidative cyclization, and we wanted to utilize this for the cyclization in this reaction. So when we went for the microwave irradiation, then we were able to synthesize this aminated teraryl, which was further utilized for the synthesis of different type of compound. And most interesting, which Sally has explored later on, that if we use the dioxane instead of the DMSO, then we used to get very good yield. So earlier you can see that it was 60-70%. Just the change of the solvent gave the yield from 60-70 to 90 plus yield. So this is the different compound which we have synthesized. We have synthesized large number of derivatives using this. Even with the fused one, this reaction was also carried out, and we were able to synthesize this dihydrophenanthrene functionalized with this amino and boron bromo compound. And this was also used to synthesize this. You can see that the normal carbazole as well as the fused carbazole, that is class of helicine. If you aromatize this fall in the class of helicine, that can also be synthesized just by using this methodology. So now I will conclude this everything. And as a researcher, like now there is a funding crunch also, and there are many aspects. So definitely, if you start some chemistry, you should try to explore everything. And here I am just showing that just using this cyanomethylarene, just by changing the reaction condition, starting material and everything, we can end up to the different type of chemistry, and all of these molecules, they are biologically as well as material properties has been explored, and they are giving very good property. It's not only the medicinal as well as the material properties has been explored for this compound, and this is the acknowledgement to all of them. So usually the material study is done by Professor Rajneesh Mitra and Rajiv Gupta. For biological studies, this Gaurav Raj Dwivedi, Sanjay Kumar and Dharmender Yadav, he was involved in the biological study, and he is the crystal expert. So usually whatever the crystal issue is there, he used to solve it. These are the students who has already done the PhD from our group, and basically this work has been done by Dr. Surjit Singh, Rahul Panwar and Dr. Sally was the main contribution of all of the work which I have presented, only these three, and now this Ashish is also exploring this work, so these are the things. And funding agency, definitely, without their support, we are not able to do this. And at last, but not the least, I acknowledge University of Delhi, who is supported by funding, by instrumentation, and by everything, and that's why we are here and we are presenting our work here. So now, thanks all of you, and if there is any question, I will be happy to take it.
Now session is open for question and
Yeah, excellent work. I am wondering, so you are using cyanomethyl benzonitrile. Okay, so if I use nitromethyl, okay, so that this kind of reaction will carry out.
Yeah, nitromethyl can be used, but then there will be limitation, because here that cyanide is also one of the very good group. Like you can see that in benzoesquinoline, that cyanide was also involved. But there was one difference with the nitro group, because sometimes nitro group also can be denitrated during the reaction. Even when we are performing some reaction with, I must say, 2-nitrobenzyl cyanide, so till now we are not able to find out that what is the structure of that compound, and we have to stop that scheme because that nitro group is going out from that substrate.
Okay.
So definitely it can be done, because that will again be a nucleophile source, but that cyanide will not be available anymore for further chemistry. And if you are putting other cyanide or other kind of group in the aromatic ring, definitely you can
And
explore it.
in your aromatic ring, you have benzonitrile. So
Yes.
if in case of CN, if I use alkyne, okay, or like property alcohol kind of thing, whether that will work
I
or
think that will work excellent, and definitely you can explore it, but I think that is very tedious starting material to afford, because you have to make that starting material. It's not commercially available.
Thank you so much, sir. I request Dr. Shelly Rana to felicitate Dr. Ramendra Pratap.
I would like to invite Professor Kestrinath Tiwari because he is the senior of my sir.
So I must say that I have many
And
senior
and
junior
junior is
students.
Dr. Vishnu Maya Desai. Ma'am, please come join us.
So she is my junior, he is my senior, she is my student, and it's really great that
So in the same boat, I would like to include two more juniors, Dr. Amarendra Singh and Dr. Ravi Pratap Barnawal, so that we make the picture complete. Please come on.
बहुत बड़ा achievement है कि इतने सारे there are compound situations, right? Even the sensor of so not there are some compounds that decided that lead towards the bacteria and I believe they are light yellow and yeah, definitely, mostly they are color. Because if they are yeah, because there is donor acceptor and you can see that there is both donor and acceptor they are also providing this application. If there is a configuration, there is possibility for
Thank you so much. Now I request Dr. Subash Ghosh to felicitate the chairs of this session, Dr. Jankiram Vaitla and Dr. Shelly Rana. We can now proceed for a tea break, but everybody has to be back by 11:40. Welcome back, everyone. Let's begin with session 11, for which I would like to invite our chairs, Dr. Amrendra Kumar and Dr. Ramendra Pratap.
So now I would like to introduce our next speaker of the session, Dr. Gori Shankar. And currently he is Associate Professor at the Department of Applied Science, Haldia Institute of Technology, Eye Care Complex, Haldia. And Dr. Gauri Shankar, he earned his PhD from Jadavpur University in 2014. And his work was based on the NHC's N-heterocyclic carbene chemistry. And he also worked as the senior chemist as MCPI Haldia since 2003 to 2011. And then he was lecturer at GIST Haldia before joining this HIT in 2015. So basically, his research was focused on NHC complexes, organometallics, catalysis, antibiotics, anti-cancer agents, gas absorption, and computational chemistry. He was recipient of awards like Distinguished Young Scientist Award and Indian Chemical Society Excellence Award. And he is a life fellow of FISC, FIC, and FRESI. He is Vice President of Haldia Vigyan Parishad and Treasurer of Indian Photobiology Society. And he published 125 publications till now and 25 book chapter, 8 patents. And he is associate editor in some of the journals. And today he is going to talk about chitosan integrated polysaccharide hydrogel fluorescence response for bioimaging. So now the floor is yours.
Very good noon to all of you. Am I audible too? Yes. So, as I'm thankful to Professor Jadhav and Vishnu Mahadi for inviting me here and all the organizing member for the last 2 days. Very good host and all the good foods and all these things. And we are very close to the lunch session. And obviously, I have a flight. So, we have to manage the time. And you know, always the organizers face this problem. And as a society member, I always organize so many seminars. So always the time is a very constant. So looking into that, last 2.5 days, you are all hearing about some organic chemistry and all these things. I have some slides on that also. And from the title, you can also understand that I am going to say something about the gel. But I have changed some plans because in the very first day, there was a debate that whether the women can do something or not. And I think from that point, we can discuss something which will be very much new to you. And I am very much happy that Pawan, my friend, he has discussed something which is not in this the scientific discussions, and it creates some interest. So as a chemistry people, we also, my general field is in heterocyclic carbene complex, as chair has already explained you. But nowadays I am getting a project from DST. I have little bit of shifted to gel synthesis. And so I will go very quickly to that. Gel, you know, this has very wide range of applications, and they have very good properties and water swelling, and basically they are used for two major purpose. One is the drug delivery in the body, or to it is the treatment for the healing purpose of any wound. And nowadays, the recent things comes, that is the corrosion inhibitor. So as I was the chemist in senior chemist in the industry, so I am little bit of interested on that. But those the gel we have prepared, that not behaves like that. But it is a very interesting case for the students in front of us, because nowadays the government job is very scarce. So you need to think about the industrial job. So you need to not only focus on the drug industry, but for the general chemical industry, where the anti-corrosive agent and the paint is a very good alternatives. So these are different kinds of polymers and these gels which you can prepare in your laboratory depending on different kinds of precursors. So what we have done, we have taken this cresol and then functionalized it to metaposition to aldehyde and then mix with the quinolazine and makes an intermediate and then mix with the ketogen and then form a gel. So I am going very short in that is discussion on it. So then if you prepare a gel, then you need to characterize on it and so many IR, NMR and all these things you can do. But the first thing you need to do is the rheology test, where you can get the idea whether your gel is, whether your compound is behaving like a gel or some amorphous compound like that. So from the geology test, what we have found that the compound has very less viscosity and high elasticity. So we can use it as a gel or you can call it as a gel. So I am not going to the detailed study of that because I have already said that I have discussed something new. So these are the, the particle range, generally the 500 to 1000 nanometer range, if the particle size is there, they generally form the gel. So this is the same image and that is the DL study. From here you can get this and all these things you can study in your Google or Wikipedia. You will find out what is the DLS, how you can do it. So I will not discussing all these things. I have intentionally hide some slides. And when two compounds are mixing together, in the definition of sol and gel, there are generally two types of compound and it's mixing together and form a new types of compound. So what we have done, we have taken the blue hydrogen and the green, this intermediate what we have found, and then mixed together. And in the left hand side, you can find the gel where you can see the two compounds are dispersed together. And the mixing is very well. So then we studied the fluorescence because in our laboratory we have tried to apply it in different field. So first target was to drug delivery. So we studied the bioimaging. For studying the bioimaging, we have studied the fluorescence. So what we have studied that in solid, it only gives a fluorescence in nearly 545 nanometer. But if we mix with water, that is when the soiling is there, that is the water is absorbed, then it will give you two peak. One is nearly 442 and one is 530. It means that is in the intermediate there is a OH and a N. So this hydrogen can transfer to keto to enol form, and that we have confirmed in the DFT study. So from the DFT study we have we optimized the two that is form, and from that we got it. Rather that to get whether this hydrogen bonding is happening or hydrogen transfer is happening, we studied the MEP, and from the red zone you can find out that this scattering of these blue points that indicates from the software that the hydrogen is transferred from the OH to from the NH, and that's why these two peaks observed. So now these are the pH dependent and the temperature dependent that we generally do in the this kinds of gel when we want to apply it, because if you want to apply it in the drug system, then what is the inside pH level of the body and what is the outside, that we need to study. So I am not going to detail of it, but the finding from here is that if we just transfer from water to THF, then the more polar to the less polar, then the behavior of the compound is differently changed. So these are the this light zone. From that, the 0% when there is the only no THF is there, the color is like this. But when the 90% THF is mixed, the color will change. And from there, these things that if you change the solvent, then you can use the same compound for the different types of imaging purpose. And this is the fluorescence which I have explained you. And this is where when it is with water and when it is with THF, what will be the aggregation pattern, that you can get the idea of it. Now these are the the half-life period that we generally calculate, or the lifetime we generally calculate. So when the water is there, the lifetime is little bit of less. Then when the THF is there up to 90%, the lifetime is little bit of increased. So depending on that, we can use this drug, use this molecule for the different drug delivery. And when we studied the temperature, then 10 to 20 to 30 degrees centigrade, the aggregation is something different. But to 30 to 35, there is an increase in the intensity of the fluorescence. It means that the aggregation from the hydrogen transfer not happening, rather this intermediate which you have formed with the quinazolene intermediate, that is aggregating together. And again, when you are increasing the temperature, that aggregation is breaking, and again the same types of this amorphous types of nature is coming, and that is giving you the different kinds of nature. Then we studied the cell imaging. And from the graph, you can find that is 0.25 milligram per milliliter is the viability data that these kinds of that is concentration you can use in our body. And going to the solvent, if you use the water, then it will only attack your nucleoside. And if you use the THF up to 90%, then it will image both the cytoplasm and the nucleus both. So the same compound, depending on the polarity of the solvent, you can use for delivery of the different kinds of drug. Whether you want to attack the drug either in the outer surface of the cell or in the whole cell. So these are the thing, and then we obviously tried with a known drug, the curcumin, and it helps us. It gives us a very good result. That in the pH nearly this 7.4, so it gives a good result. So that is all about the chemistry. As to manage the time, I have skipped this part. Second part, that is now I am going to describe you a love story. So the love story of a woman and who make the change of the scientific community of India, and probably most of you do not know that. And that starts with a Bihar lady. Her name was Professor Rohtagi, K.K. Rohtagi Mukherjee. Full name was Krishna Kamini Rohtagi. Not Mukherjee. Mukherjee I will come in later. So Krishna Kamini was a Kshatriya in caste, and in India casteism is a very big problem. So she faced the problem very big. So Krishna Kamini was from Bihar and came to Calcutta for the higher studies and fall in love with that time a very great chemist Sushil Mukherjee, and they had a very long time love. But what happened? Sushil Mukherjee was from a Brahmin, and his mother did not allow to get a the bahu in the home who will be the non-Brahmin. So their love was stopped there, and Sushil Mukherjee, as a chemist, has a wider application throughout the world, and he went to postdoc also, gave so many institutional heads, and in 1976, he came to Calcutta University and joined as a vice chancellor. And at that time, his age was 58. So this time is very much important, right? For the younger students, I am mentioning the age. The age was his 58, and in 1978, he retired from Vice Chancellorship of Calcutta University, and his age was 60. And that particular year, Sushil Mukherjee's mother died. So there was no restriction to get married again. So Sushil Mukherjee married to Krishna Kamini, and Krishna Kamini Rohtagi became Krishna Kamini Rohtagi Mukherjee. And for the biological cause, they cannot give birth to a child, so they have no child. But what happened is that during this time, Krishna Kamini Rohtagi Mukherjee did something. He wrote a very beautiful book, and till date, I am again repeating this word, till date, this is the only book in India which is the fundamental photochemistry book. And if somebody has any interest in photochemistry, then only this book is available, and you can call it as a Gita of photochemistry. So the left hand side, the upper side, she is the Krishna Kamini Rohtagi Mukherjee, and along with some of the scientists, she established a society in 1964, which is called Indian Photobiology Group, not the society. But in 1970, they registered it, and the group became named with Indian Photobiology Society. And that time, these people helped him a lot. Helped her a lot. So these are some big names of the photochemistry or photoscience or photobiology, whatever you call it. And yesterday, one I think Sudeep Basu, I met with him, and when I invited him to give him a lecture in the next April, he asked me that the institute name is Indian Photobiology Society. So what is my role? So again, I am coming to that. That if there is a woman in particular institution, what she can do, what possibly the men cannot do. The problem is that generally the men are egoistic, and the women are less egoistic. So they can accommodate more people, right? So what she did, you will be surprised from this. This is, this is the first executive council established in 1970. And obviously, Vice Chancellor Sushil Mukherjee having that time the DSc degree, not this time they paid 5 lakhs rupees to any university and they will give you the DSc degree, not like that. So getting a DSc degree was a very tough process. Till now, getting a DSc degree in a regular way is a very tough process. That is something different history. So he was the president of that. He is from chemistry. Then N.N. Dasgupta, he is the first person, he is the first person to establish the single ion mass spectrophotometer at Calcutta, and he is the physics person. Then the third one is from Department of Botany. Then the fourth one is from Department of Applied Chemistry. Then the fifth one, G.V. Joshi, is a very famous name, If you search the literature, G.V. Joshi is a very famous name. Because nowadays we do not culture about the history of the chemistry. That is our problem. So he is from botany. Then K.K. Rohatgi Mukherjee herself is from chemistry. She was a physical chemist, obvious. And B.B. Biswas are from biochemistry. So these are the executive council different positions. And now look at to the right side. These are the members. And B. Mukherjee, I don't know his field. Probably he is from physics. And Deepesh Ghosh is from botany. Anjali Mukherjee is from environmental science. And J.P. Mittal, he was the youngest member that time. And he was giving last time we gave him the Lifetime Achievement Award and he gave us a story that he was giving a lecture like the students giving the oral lecture today. So he was giving a lecture and Rohatgi Mukherjee was the audience. So she was so impressed that immediately he called him to join the executive council. And J.P. Mittal is the youngest member that time and now he is nearly 70 years of age. J.P. Mittal from BARC, everybody knows that. He is a Padma Shri scientist also. And the next one is a very impressive name, K.M. Panikkar. That time he was the #1 oncologist, cancer specialist in our country. So he is a doctor. Now proceed with that biophysics division for S. Joshi, A. Kumar and V. Ramakrishnan, everybody knows. He is a good chemist. So this is what, I took the charge as a treasurer in 2022. And when we took the charge, I am not a biophysicist, not a biochemistry, not a Indian photobiology or like that. I am simply doing the chemistry and but I have a little bit of experience with Indian Chemical Society for nearly 6 years as an executive member of there and handling some of their publications as an editor. So they pick up their me to this society as a you can say as a what you can say as a influential person or like that, or I work hard like that. And when I joined there, the registration was not there for 5 years. There are no documents. So we need to re-register it and to restart the society again. And the interesting part is that the Journal of Photochemistry and Photobiology wrote about the Indian photochemistry history in their own journal. And they wrote a special paragraph of Indian photobiology. And if you go to that, all the big names will come. Right. So next is, this is our present executive council, which is started in 2025, and Professor A.K. Mishra, Ashok Kumar Mishra from IIT Madras, he is our now president. Nitin Chattopadhyay, who was the past president, now is the vice president, because in our rule there is the past 2 president will be the 2 past vice president. Amitabh Chattopadhyay has recently joined with us. Everybody knows him. He is the CCMB founder director. Vandana Bhalla, she is also a famous name in the women scientist category. Chitranjan Sinha is a very, energetic person and everybody knows him because of his energy and societal activity. Myself is the treasurer. Aninda Datta from IIT Bombay. Kishore C.S. Panigrai from Niger Bhubaneswar. Kolol K. Ghosh from Pandit Ravi Shankar Shukla University. Rifat John from Jammu Kashmir University. Jhuma Ganguly from IIT Sipur. Chandrabhas Narayan is now the director of Tiruvananthapuram RGCB. Alakesh Visai, yesterday he was here, IJR Kolkata, and Saptashi Mukherjee from IJR Bhopal. So these are the new council from 2025 and it will act up to 2027. And what is our activities? That is like this. So every month I will inform the students and I will also write a letter to the head of the department to this university that every month in the last Saturday in the evening 7 to 9 p.m. we organize a monthly talk and luminous scientists from India and abroad, they give a talk. And when we were in Indian Chemical Society, we started this. And I can claim that very proudly that ACS starts this when Indian Chemical starts that. So Indian Chemical Society starts this science lecture first, then ACS started. So we are that pioneer. So now Indian Chemical Society do not do that, but Indian Photobiology Society we have started it. So we have a newsletter, IPS newsletter, but from this year we will change it to Proceedings of the Indian Photobiology Society because our president wants that this will be converted to a journal. So we are trying to make it, and we organize two big program. One is the Foundation Day, which is the 9th July, and 2nd August. Incidentally, sometimes happens, which is incidentally happens, right? Accidental, incidental, like that. So Rohtagi Mukherjee birthday is 2nd August, and the birthday is 1924, and PC Ray birthday is 2nd August, and he established Indian Chemical Society in 1924. So when Indian Chemical Society established, Rohtagi Mukherjee was, he came to this earth. So it is a coincidence maybe. So going to that, on 2nd August, we generally organize a big program. And within this July to August, we organize an international conference. And last time it was in KIT, and the Foundation Day organized in Aijar, Kolkata. So probably this time it will be in JNC, sir, and Chandrabhas will look after the matter. And these are the awards which we have started. Previously, only one award was there in the name of K.K. Rohtagi Mukherjee Memorial Lecture Award. But now we have started so many lecture awards with the all luminaries who was in the Executive Council. Along with that, last year was our Diamond Jubilee. So we've a special award give to one scientist and also one philanthropist who helped the chemistry study. So that's why Achyuta Samant also got a award. And Kitagawa, everybody knows that he is the father of the MOF. So he got a award. So these are the different kinds of award we organize. And in every August, as I have told you, 2nd August, we celebrate the birthday of K.K. Rohtagi Mukherjee and our god of chemistry, Acharya Prafula Chandra Ray. And this is our different kinds of event, the monthly science talk or like that. Okay, I'll complete it. These are the different posters or flyers which we generally circulate during all the monthly talk. And sometimes we organize some special talk when in memory of some special scientist. So that we have organized in several times. And Kankan Bhattacharya is one of the big name in food biology. And last time this program was organized. And this is our collaborative activities, that is in different college we generally go and organize with them. We support with them. Also in this conference we are supporting the organizers with 5 poster awards and like that. So though it is not a big amount, but we are trying to collaborate with this. And we try to make some students chapter in different college, and we generally encourage the students, even the that is undergraduate students, to participate with the poster, such that they can get an idea what the science is going on. So this is our previously IPS newsletter. It was a print version. Now it is in the online version, so you can get it from our website. But from this year, it will be converted to Proceedings of Indian Photobiology Society. These are the program which was organized in Aijar, Kolkata. A little snapshot. This is the program in KIT, and these are the different kinds of. And this is the awards which we have given some of the big names. And one of them is, that is J.P. Mittal received the Lifetime Achievement Award. And that's the Pradeep Sengupta, he was in SIMP, he got the Lifetime Achievement Award. Kitagawa get the Jubilee Award, Achyuta Samanta get the Jubilee Award, and so many names is, and the photo is there. So this is the membership form. So if somebody wants to join with us, especially the faculties, you can join with us in the live membership of 3,100 rupees. From July onwards, it will be 3,500. So if you want to save 400 rupees, so you can join within April to July. And for students, you can join with 300 rupees. And if in your institute there are more than 20 students and you want to form a student chapter, then from this 300 rupees, we will again give back you 150 rupees, such that you can start your student chapter. So basically 150 rupees will go to society and 150 rupees will come back to your institute again. And similar to SCAS or RSC, the student chapter you can form. One student will be president, one student will be secretary, one student will be treasurer. But condition is that your institute head should have the permission, and one teacher will guide you. So with this, I must thank the audience that you have given some time to listen this societal activity. And when I start this presentation, there is a picture in the left hand side. Probably you have not care about that. And again, I have repeated this. This is the PCRI picture, and this is drawn by my elder son, and that is been taken to the particular journal for the cover page. So nothing to clap. What I am giving you the interest is that you can earn some money from that. So what the point is that what Prabhu was trying to say, that you can transfer the copyright and get some money. So my son also get $100 from this picture. And similarly, ACS got received another photograph of that. That is also drawn by Hilderson. And in both this case, I was the one of the editor. So it was a little bit of helping like that. You can say that father helped the child. So anyway, he received there from $25. So if you have some interest in painting, you can contact with some of the journals, because nowadays the journals are asking for the cover page, not from the authors, even from the outsiders also. So with this, thank you very much thank you very much for listening to me, and I'll be happy to meet in any future program, because due to the shortage of time, the chemistry section I have to skip. So for that, I apologize. Thank you very much.
Gori Shankar for the information of the society as well as your scientific part. So if there is any information you required regarding this or scientific part, you are most welcome to have a question.
I think we are going shortage of time. So if anybody has any questions, my email is available, and I am always available with my WhatsApp also, so you can connect with me. Better to call the next speaker.
Definitely. So thank you.
I request Dr. Ravindra Pratap to felicitate Dr. Gori Shankar Roy Mahapatra.
The next speaker of this session, Dr. Santanu Gosh is Assistant Professor at Department of Chemistry, Shiv Nadar Institute of Eminence. Dr. Santanu Ghosh is an Assistant Professor in Department of Chemistry, Shiv Nadar School of Natural Sciences, and he earned his PhD in Organic Chemistry from ISER Bhopal under the supervision of Professor Alakesh Bisai. He then pursued postdoctoral research at the University of California, USA with Professor Zakarian, and he also did another postdoc at Max Planck Institute, Germany with Professor Benjamin List. His research focuses on natural product synthesis, asymmetric catalysis, and new reaction discovery. So, with this brief introduction, I invite Dr. Santanu Ghosh. The class is yours.
Thank you very much for your kind introduction. First of all, good afternoon to all of you. And my special thanks to the organizing committee, particularly Professor Visnumaya Visai, for inviting me in this beautiful audience. Yeah, today, yeah, I started my independent career just few months. I mean, it's about 2 years, close to 2 years. And today I'll be talking about my recent research contribution to the scientific community. And I will begin with my postdoctoral research, and then I will come to my independent research, what I started with. Let's begin with that. I worked with Professor Benjamin List with a concept which is asymmetric counter anion directed catalysis. Actually, it is a powerful concept in organic synthesis. So if you are in asymmetric catalysis group, or you want to develop a lab based on asymmetric catalysis, so you have to know two things: how you are going to activate your substrate, that is called activation mode, and second thing is that how to create a three-dimensional space so that you will be able to form a enantioselective carbon-carbon or carbon heterocyclic bond forming reaction. Well, if you go to the literature, what you will find, there are mainly two different types of activation modes are available. First one is called classical covalent activation mode, which is nothing but enamine catalysis. Yesterday, we have some lectures based on this enamine catalysis. Another one is nucleophilic catalysis. The second activation mode could be classical non-covalent activation mode. What is that? Mainly it's depend on the hydrogen bonding catalysis or ion pairing catalysis. With the help of these two activation mode, now I am going to introduce the term ACDC, which is asymmetric counter anion directed catalysis. Probably you have learned in your BSc that phase transfer catalysis, that is nothing but a formation of a ion pair. Here also in the inside this concept, what is present, actually, if your catalyst is there, catalyst may have counter cationic part and a counter anionic part. Now the counter cationic part, what it can do, it can activate your substrate. Substrate will convert it to the substrate catalyst pair. Then that will be a cationic pair, and the remaining part will be the anionic part. If you have some chiral information on this anionic part, what it will do? It can form a ion pair, tied ion pair. Because of the tied ion pair, there will be a three-dimensional space. And due to the presence of this three-dimensional space, what you will be getting? The nucleophile or electrophile, whatever you are going to form the bond, mainly here will be nucleophile, that will come to a particular phase and form the carbon-carbon or carbon heterocycle bond. And that is the actually principle of ACDC, and it was coined in 2006 by Professor Benjamin List. Utilizing this concept, and we use a very strong and confined acid. This is a just representation of, shortest way to represent that strong and confined acid. We have done several transformation. I was mostly focused on the 4+2 cycloaddition reaction. You know that 4+2 cycloaddition reaction is very valuable reaction to construct multiple stereogenic center in a single step, and also has, you can create a quaternary center as well. Utilizing that concept, we have constructed a decaline motif having a 4 to 5 stereogenic center in a one step. Again, we have constructed a spirocarbocycle. You can see that spiroquaternary center adjacent to a non-stereogenic quaternary center. All are challenging reaction because we are going to construct the quaternary center, and that too in enantioselective fashion. Further, we employed our strategy for the construction of lactone motif having a quaternary, means tertiary alcoholic center, and that followed a protolactonization strategy. We also employed this strategy, this 4+2 cycloaddition strategy, for the synthesis of beta santalol, which is the key component in sandalwood oil, whatever you are using every day for fragrance or some other purpose. The fifth one, this is also a synthetic arboron, actually fragrance compound, which has almost 5 picogram per liter, that is threshold limit. So let's start with. I will today I will cover mostly this synthesis of this decalin motif as well as pyrocarbocycle, and then I will start my independent. I will show you my independent research career, research topics. So here what we have done, if you see the literature, there are plenty of molecules available that contain the decalin motif, densely functionalized decalin motif, and you will find a quaternary center present at the alpha position of the fused ring system. That give you extra challenge to construct them in enantioselective fashion. What we thought here, we can utilize this 4+2 cycloaddition reaction using a cross conjugated dione, and in the presence of diene we can construct this decalin motif with 5 stereogenic center. So if you have some knowledge on stereochemistry, immediately you will come to know that 5 stereogenic center means you are going to generate 32 stereoisomers, right? However, since we are doing the 4+2 cycloaddition reaction, we are taking the advantage because Diels-Holder reactions are proceed via the suprafaciality as well as it will maintain the stereospecificity of the process. That actually reduce this 32 isomer to the 8 stereoisomer. Again, cycloaddition reaction proceed via endoselective pathway, that again will reduce the total number of isomer from 8 to 4. Now our job is to control this 4 diastereomer to a single diastereomer by using our catalyst. However, the enantioselective process will be challenging because your diene, if you see that, except this carbon, all are sp2 hybridized. That means rest part is flat. It's kind of a planar part, except this part. Since it is planar, diene can approach all around the circle. It can come from the top, from the bottom, this arm, or pro S arm as well. Furthermore, the activation center means activation part active part of this dienophile is CO, which is far away from the quaternary center, which is called remote quaternary center. Because of that, it will be really challenging to control the diastereoselectivity of the process. Okay. Again, if your RL and RS substituent are sterically unbiased or electronically unbiased, almost similar electronic and steric configurations are there, then it will be very difficult. So how to control that? What we hypothesize that to overcome all these limitation, if you can somehow generate a highly acidic and confined catalyst that can have the opportunity, that can give us the opportunity to control all these difficulties associated with this transformation. Why you need the high acidic catalyst? You know that catalyst has a role to reduce the activation energy barrier, right? So here the acidic catalyst, if your acid is strong, it will donate this proton and reduce the LUMO energy of the dienophile significantly. And the confined catalyst is required because after the transfer of this proton, it will generate the cationic complex. Now it will enter into the confined cavity of the catalyst. When it is entering into the confined catalyst of the confined space of the catalyst, there will be only a particular space available for the diene to interact with the dienophile, and as a result of that, you might have the opportunity to get the good level of enantio and diastereo control. With this concept, we have started to develop the process, and here you can see the term I use, sterically and electronically unbiased. See the methyl and ethyl, very minor difference in sterics and electronics. We utilize this substrate to optimize the process along with this cyclopentadiene as a diene source. And again, the acids, different sets of chiral acid we utilize. You can see the pKa value is mentioned below to that acid. You can see that when we use simple phosphoric acid, reaction did not work. This modified phosphoric acid, which is called imidodiphosphate, again reaction fails to produce any product. Then increasing further acidity to disulfonamide, reaction failed. Further, we got some result when we utilize this imidodiphosphoramidate. The beauty of this catalyst is that you can tune up the catalyst confined space as well as acidity. If you changing the R group, you can actually increase the acidity by introducing the electron withdrawing group on this inner core. Furthermore, if you change the 3,3' substituent, you can actually tune up the cavity of the catalyst. to our delight, we can able to get the good level, good to excellent level of enantiomer control, and we have the optimized condition utilizing this particular catalyst. I am not going to into the details of this catalyst structure, but you can follow the paper indicated over here. And we have synthesized several sets of substrate. Here few of them I am representing, and we also converted this substrate to the some of the valuable derivatives, such as you can see that we can construct a quaternary center at the delta position of this ketone, gamma position at this ketone, and also one can do 2+2 cycloaddition reaction to generate this cage like structure and many more. With that, I want to move to the second part, which I want to discuss how we have constructed this spirocarbocycle. If you see the literature, there are plenty of natural products available that contain actually this kind of spirocarbocycle. Few of them I am representing here, such as acrone, spirovativen, or omphalen. Can you read this? No, this. Can you? So omphalen and also chemigreen natural product class. All this class is having this pyrocarbocycle, which is very congested environment. Even sometimes you will find that this pyrocarbocycle is adjacent to a non-stereogenic quaternary center. That is giving you extra challenge how to develop the method. If you see the literature, so all the enantioselective methods available. So for two, I think before we publish this result, there are only two literature available, and they follow a multi-step protocol. What we thought, if we can use a exocyclic enone that can do a 4+2 cycloaddition reaction and can generate this pyrocarbocycle in a single step. However, this kind of reaction will be challenging. So you need to think about two things if you want to get enantiomer pure compound. First, how you are going to reduce the LUMO energy of the dienophile. To do that, you have to generate a highly acidic catalyst which can coordinate with this means exocyclic enone and reduce its LUMO energy. And the second thing is that again the confined geometry of the counter anion, so that after the activation of this dienophile, it will enter into the confined cavity of the catalyst, and the catalyst will give you only one particular phase to approach the diene and give you the enantiomer control of this process. With this concept, so these are the requirement I already mentioned to you. With this process, we started optimizing using this diene is called myrcene, and this is the exocyclic enone, and we are happy to observe that again we are happy to observe that yes, our hypothesis was correct. We need very acidic catalyst, and again this beauty of this catalyst, we can tune up this 3,3 prime substituent to tune the cavity of the catalyst, and we had optimized condition using this when this air is para pentas means SF5 compound aryl group present over this 3,3 prime position. This is called 3,3 prime, and in the inner core it is having C6F5 pentafluorobenzene. With this, we explore the substrate scope of this method, and few of them I am representing over here. We also extrapolate this methodology for the kinetic resolution. Actually, this is the material used for the synthesis of choletoic acid. Furthermore, we also utilize this enantiomer pure compound to the corresponding beta chemigreen via a simple Wettig olefination reaction, and followed by the methyl lithium addition to this ketone and triplet, followed by cross coupling with methyl magnesium iodide to generate this alpha chemigreen. Furthermore, this is actually was utilized for the racemic synthesis of omphalic acid. So this could be a viable procedure for the synthesis of enantiomer pure omphalic acid. Again, we also utilize for the synthesis of Lawrencian on C, where we followed a alpha beta Sagusa-Ito condition to generate this enone, followed by methyl lithium addition and transposition of this OH, followed by oxidation. So, as an independent researcher, the field I have chosen, that is the synthesis of bioactive natural product and catalyst design, as well as in the asymmetric catalysis field. And our lab also actively working on the generation of new reaction methodologies. So current target, we have chosen this asper Novin molecule, which I got the DST grant, SRG grant. And I am also working on this chroman type of natural product. Furthermore, our group is also working on this NIQ natural product, naphthyl isoquinoline alkaloids. And these are the current research focus I have written over there. Let's begin with this naphthyl isoquinoline natural product. So here, why we are interested? You know, the malaria is a global health burden. In 2023, World Health Organization showed that almost 263 million clinical cases and including 5,97,000 deaths. However, if you see this graph, this India is over here. India also having lot of malaria cases, and all this red zone in this world map are actually malaria prone region, whereas the green region are the malaria free region. So, however, you have to know what is the cause of malaria. The main cause of malaria is the two key protozoa is uh, Plasmodium falciparum and Plasmodium vivax. The malaria actually can be cured by utilizing this chemical composition name is artemisinin, which was discovered by Professor T.U. Because of that discovery in 2015, she got Nobel Prize. However, this Plasmodium falciparum actually muted, and due to the mutation, it is actually drug resistant. And because of that, scientists are in search for a new drug molecule. If you see the literature, you will find that this kind of naphthyl isoquinoline alkaloid actually showed very promising biological activity. Even dianocophylline C and zogiamine showed the best activity. You can see the IC50 value is almost 0.0014 micromolar. So from where one can get this molecule, so that's why we are targeted this molecule. And from where one can get this molecule, one can get this molecule from African and Asian lian trees. Now let's see what are the difficulties associated with this synthesis. You can see that you have to maintain a mild reaction condition to preserve this axial chirality, because okay, I'll finish it up. Because if your reaction condition is drastic, it may get racemized during your transformation. Furthermore, what are the other difficulties? You are going to construct this bond when you will have two aryl ring having tri-ortho substituent. That is really challenging. Another thing is that all the naphthyl ring or the other phenyl ring that are highly functionalized, we have to prepare those also. In this direction, what is our hypothesis? Our hypothesis is that can you utilize a desymmetrization strategy where you'll have the symmetric molecule, both the side is having O triplet, and now if you can do a asymmetric palladium zero catalyzed coupling reaction with this nitroolefin, then you can immediately generate this enantiomer of your intermediate. Upon hydrogenation, you can generate this amine. Now, simple Bisler Neprileski or Pictet Spengler cyclization can give you this kind of naphthyl isoquinoline alkaloid. However, my students tried lot of reaction condition, but they could not handle this nitro compound. So I have to revise this strategy, and what we found that we can actually use this acrylate derivative where you can easily construct this bond. For there, after that you have to do the hydrogenation followed by methylation and aminolysis. You will reach to this amide, and then you can follow a Hoffman degradation to generate the amine, and then you can follow a Pictet Spengler cyclization to complete the synthesis. So as we hypothesized, we started working on this. After several screening, we found that yes, that can be done. This is the racemic substrate scope. Enantioselective scopes are going on. Probably we have some promising result. Probably it will take few more months to complete that. And as we have already completed the racemic synthesis of our desired product, we are interested to convert to the corresponding natural product. For that, we have done this detripylation followed by hydrogenation of this double bond, and then methylation followed by aminolysis. We have now this amide structure, which we are now working on this Hoffman degradation step to convert this amide to the amine, and again the cyclization will give us the this desired natural product. With that, I want to move to the most important slide in this talk, that is the acknowledgement. Without this slide, I'll be this talk will be incomplete. I want to acknowledge all of my supervisor, Professor Alokesh Bisai, Professor Armin Zakarian, Professor Benjamin List. Without their hard work and sharing the knowledge, I will not be able to start my independent career. So along with that, I also want to acknowledge all the institutions I have been associated with, that Vidyasagar University, ISAR Bhopal, University of California, Santa Barbara, Max Planck, CSIR for fellowship, Eli Lilly for AC Outstanding Thesis Award, and IUSSTA for postdoctoral fellowship, and my current organization, Sivnadar University, DST for funding, and my PhD student, JRF and MSc student. Thank you very much for your attention. And if you have any question, you may ask.
The floor is now open for queries.
2023. Yeah.
Okay, so the last molecule, whatever you are doing,
Yeah.
isocon, that this thing. So why you were making the racemic one?
Because enhanced selective process also going on.
Going
So I have to make the, first you have to synthesize the racemic compound, and then you have to separate in the HPLC or GC, wherever it is, it can get
the
separated.
molecular enantiomer or diastereomer?
Which one?
Go to the last slide.
Yeah, that's diasteo.
That's yeah, that's
That is
diasteo. So you're talking enantiomer. So
I am talking about.
So no, last next one.
Yeah. That is diastreamer.
That's that's a diastreamer. Yeah, that's something.
That is diastreamer.
So you are not able to control anywhere the stoichiometry.
Not yet.
Not.
The work is going on.
Okay, very good. Excellent work. Yeah.
Thank you.
I have a one question. In your first case, you have shown that you have used only cyclopentadiene as a diene.
Yeah.
So why you don't used any other dienes like valence diene or electron rich or acyclic diene? Because in the second case, you have shown that you have used acyclic diene like butadiene.
Yeah, I have used, but I put only few.
Because in all the cases when we synthesize this cyclic the decalene system, there you have one appendage of the CH2 bond.
Yeah.
So
I did not show over here in this slide. Time region is only 15 to 20 minutes, right?
But
yeah, that is there. You can follow that paper.
Okay.
Thank you. You can get it.
We'll request everyone to catch him later during the lunch time. And let's thank Dr. Santanu Gosh for a fantastic talk.
I request Dr. Amrendra Kumar to felicitate Dr. Santanu Gosh.
So now our next speaker, Dr. Navita Sadukhan, is going to present her work, and currently she is assistant professor in ICT Mumbai, and she is specialized in biomimetic chemistry for functional, colorant, and specialty chemicals. She has got her PhD from IIT Kanpur in 2009, and she was faculty at Tokuho University, Japan for 4 years. Her work includes the proton transport mimics, enzyme mimics, catalysis, self-cleaning cotton, fluorescence controlled drug delivery, and artificial photosynthesis. Her self-cleaning cotton was recognized by the Ministry of Textile, Indian government, and she is recipient of Serve Young Scientist in 2014 and Top 20 Research Award by German Chemical Society in 2014. She has 25 publications, 3 patent, and many project is going on with her. And with this, I would like to invite her to present her talk on catalysis to achieve known molecule with unique theragonist benzothiazole for protein responsive drug delivery and copper phthalocyanine for anti-cancer application. So, welcome.
Hello. Thank you, Professor Chair, for the kind introduction. At the outset, I would like to thank Professor Yadav for the wonderful hospitality in this green, lush campus and pleasant stay. And I would also like to thank the organizers and Professor Vishnu Maya definitely for the invite. So as my talk title is Tegylation to achieve various functional molecules for biological application, first I'd like to introduce the institute ICT Mumbai. Some of you may know, but still for the sake of students, that this institute is located in Mumbai near Dadar, and the institute is established in 1933. I work in the department of dye stuff technology. Earlier, the name has been changed to specialty chemicals in 2021. We closely work with associated with various industries, and my specially work is to find out few biomimetic solution for technological challenges. So I work in various biological relevant work. Some of them are proton transport mimic, enzyme mimetic catalysis, lotus effect, fluorescence directed drug delivery and anti-cancer application, dye sensitized solar cell and natural light harvesting system. Today I plan to introduce you with brief discussion on mimetic catalysis, enzyme mimetic catalysis and lotus effect mimicking, and I will discuss detailed fluorescence directed control delivery and anti-cancer application. To start with, enzyme mimetic catalysis. This enzyme is called xanthine oxidase is present in liver. This specifically oxidizes hypoxanthine to xanthine form. If it functions differently, then it will result in uric acid. So the controlled oxidation of hypoxanthine to form xanthine to make the cell work functionally. What happens here, you see hypoxanthine, it has two oxidation centers where this 2 and 8 positions are prone to oxidize. So you need to control oxidation to form xanthine. So if we analyze the xanthine oxidase active cofactor, where molybdotyrine is the one which where pyrene moiety with diethylene stabilizes molybdenum dioxide that functionally oxidizes xanthine, hypoxanthine to xanthine. So to mimic this functional molecule, this molybdotyrine, various papers reported that is quite challenging, including NUN Chemi and then JAX. It's specifically says this diethylene pyrene moiety is responsible for this electronic factor that leads to the catalysis. So challenging part is to mimic this functionality using a simple molecule, which I suppose there is one naturally abundant molecule which is maltol. You can see this pyrene with this molecule can work as a dichelate anionic ligand. This can nicely stabilize molybdenum dioxide moiety that I name as molybdenum dimol catalyst. And this is one thing very significant here. You see this carbon-carbon double bond nature that leads to the electronic factor for this active oxygen transfer for this catalysis reaction. Now this in this the extra crystallography indicated that here this C1C5 bond is nearly 1.437, which is nearly double bond in nature, which leads to possible organic atomic transfer or oxygen atomic transfer reaction. Further, this catalyst was taken for this catalysis reaction, xanthine oxidase, like catalysis reaction where hypoxanthine was taken and then it selectively oxidizes to xanthine. The reaction was monitored by proton NMR, which led to this only H2 proton intensity changes that indicating the selective oxidation is taking place. This catalyst was further taken for various other oxidation reaction to make value added product. This result was published as a cover page in Dalton Transaction. Further, briefly going to the my very dearest project, which is called Lotus effect on cotton fabric. So what happened in 2020? When this lockdown announced, then I saw one of the compelling situation where doctors and nurses and medical practitioners are working inside PPE kit. So this PPE kit is actually made of nylon polypropylene synthetic material that lead to their struggling inside with excessive sweating that lead to the dehydration and various health hazards. So what I question actually that why they can't use PPE kit made of cotton fabric, which is very comfortable, breathable, and which lead to very working hour will make convenient. So when I searched that whether cotton fabric can be used for this PPE kit, please play the video, that I found that cotton fabric is actually hydrophilic in nature that will lead to absorption of this bacteria and virus and then leading to this contamination or transmission of the diseases. So it can be useful for the application. So what I need is that we need cotton fabric which is breathable in nature. However, it should have some antibacterial, antiviral property, super hydrophobic or self-cleaning property, so that it can lead to the cleaning the surface where the bacteria and viruses can be removed. And if it has, since because of this excessive sweating it has an anti-odor property, it will be added advantage. So why I saw the nature that there is a lotus effect present, by which this by definition is that in the lotus paper lotus leaf, if you drop a water molecule, droplet, so it falls through the leaf and then collecting all the dirt particle from here. So that is called lotus effect. So if that property can be created on the fabric, it will be a great application for this PPE kit. Now to design. Go to the next slide, please. Yes. No, just. When we did some literature survey, we saw that this is possible on material, it is known, but using some polymer. The similar polymer we can't use for fabric application because it will compromise the quality of the fabric and it will have this synthetic material. So the purpose will not be solved. So we thought of designing various dyes so that it can incorporate those property, antibacterial, antiviral property with super hydrophobic surface. So I designed quite a library of dyes. The champion dyes are this presented here. So I synthesized this dye, we apply it on the fabric, which lead to this antibacterial, antiviral property. So concept I am not discussing, I am just skipping a little faster here. So you see that we have developed some technology how to apply the dye in the fabric, and you see the nicely super hydrophobic 153 contact angle was observed. So which lead to the super hydrophobic. The definition of super hydrophobic is that water contact angle essentially would be more than 150 degree. So here upon dyeing, we could achieve that more than 153 degree water contact angle. So this can be useful as super hydrophobic cotton fabric. So this is the video Please play it. So here on the fabric, you see that the dart particles are sprinkled on it, and then upon applying water, it rolls off with taking away all the dart particle. And here sees you see the contact angle is quite high, and it can be used as super hydrophobic cotton. Further, this dyed fabric can be used as self-cleaning surface also. You see that upon light irradiation, the dirt, this stain on the fabric is completely removed within 6 hours by just illumination of light. Further, this molecule shows more than 99.74 and 99.02 for both gram positive and gram negative bacteria. So, and this fabric can be washable, reusable up to 30 cycles. So by this we achieved breathable antibacterial self-cleaning surface, and this work was highlighted by the Minister of Textile in Bharat Tex Expo. Further going to the talk in detail on tegylation. Now tegylation is the definition of tegylation, that if you heard of the pegylation means is a conjugation with the peg molecule to various biomolecules, protein, drugs and dyes to achieve its prolonged circulation time, enhances bioavailability, improved stability, reduced immunogenicity, increased solubility. So if we decrease this, the chain length to tetraethylene glycol and then conjugate with various molecules, that would lead to tegylation. Now, this pegylation or peg molecule has its own importance for in pharma. The peg 3350 used as a laxative. Peg can be used as a binder to improve their solubility and bioavailability. So my idea is that the pegylation which is peg molecule which is available in the market is a polydispersis nature. So if we take this peg molecule of any weight, I have taken peg 600. This is the mass spectra of peg 600. So it indicates the kind of polydispersity it has. However, that is because here this polyethylene glycol is made of the various ethylene dioxy molecule, and because of this gauge and the the conformation, this molecule gets this amphiphilicity. Now, depending upon the number of Gauss anti-ratio, the hydrophobicity varies, and it also depends on temperature, light, and the number of chain. So, if you have this polydispersed molecule, the bioavailability or the stability of the protein varies. So, what I wanted to do is that we take specific length which is monodispersive in nature. If we can conjugate with the biomolecule or any drug molecule or any dye molecule, then it will lead to very specific property. So I have chosen take as the chain length. Now, this TEG molecule, the advantage of TEG molecule is it is non-toxic, non-ionic, monodisperse, easy diffusible in biological fluid and amphiphilic in nature. It is commercially available and quite economic to use. Further, the intention is to make a drug delivery system using a small molecule. So now there are various dyes, they are very specific for very specific cell lines. Now I have chosen here the benzothiazole because it has very specific bind site with beta amyloid, and I conjugated with the TEG molecule considering the fact that this is entire molecule is neutral and it can be used as a drug delivery system for neurovascular system. Now, I synthesized this molecule up to here. It was known this part where hydroxybenzothiazole unit is conjugated with TEG molecule by Williamson ether synthesis, leading to 76% yield of TEG BTA2. This molecule was chemically characterized by NMR, FGIR and mass. Further, it was taken due to the amphiphilic nature of this molecule. This molecule indicates that various aggregation pattern in the water. So, and it also polarity with changing polarity, its fluorescence property changes. So what I did is that we have taken this molecule in THF and started the aggregation pattern with adding water. It shows that molecular aggregation takes place. This close analysis of this NMR indicated that here the thiazole unit is interacting with the dimethyl benzene unit by pi-pi interaction, leading to the molecular aggregation. Further, absorption and fluorescence spectra indicated that fluorescence intensity increasing, that is leading to aggregation induced fluorescence, that is because of this restricted internal rotation. Further, the intensity decreases because of increasing size of molecular structure lead to the precipitation of this self-assembled structure, leading to the fluorescence intensity decreasing. Further, so particle size analyzer indicated this particle size is actually 520 nanometer, and ACM clearly showed this very nicely round shape microsphere. Now, the recording CLSM, you probably may not notice the blue color because of this. This molecule is visible under CLSM. This molecular self-assembly. Now, interesting part is that when we take this molecule self-assembled in water, added curcumin, and it form this microcapsule structure. And now you see this rice-like microcapsule actually under optical microscope, and then this is optical microscope, this is SEM image, and you see CLSM blue color molecular aggregated structure. Now I find this interesting resemblance with this rabies virus, where this structure resembling like rabies virus, a bullet-shaped molecular structure, assembled structure, and this is enveloped RNA virus. Membrane is made of glycoprotein and very known for cell entry and neuroinvasiveness because of this structure, and the glycoprotein consists of the cell membrane. So our structure resemble like this, so it will be probably very good for the drug delivery. So further, I have taken this microcapsule, interacted with BSA protein, and it shows where curcumin released in presence of BSA protein, and this is the absorption spectroscopy, which lead to indicating that this molecule is getting released after adding BSA protein. And also CD recorded that this protein is getting attached with this molecule, and you see that when we added these microcapsules, and this ACM image indicated that after addition of protein, that microcapsule is disintegrating, indicating that this molecule is releasing the drug and interacting with protein. So we could prepare one system, drug delivery system with dual functionality made of tetraethylene glycol conjugated BTA2 system, which interacts with the protein and lead to drug delivery. Further, I am going to the next topic, that cancer cell specific oncological application. So here, for normal cell that we know, the various enzymes works normally, definitely. One of these enzyme is glutaredoxin system. If it starts malfunctioning, then the concentration of GSH increases in cancer cell. That lead to the pH, acidic pH to the cancer cell, hypoxic, and H2O2 concentration increases. So if we really want to design some system which will be specifically working on cancer cell, devoid of normal cell, without affecting normal cell, we really want to see this, how this parameter affects this drug acting system. Now we have thought of this unit, which is phthalocyanine unit, is known for photodynamic therapy to kill bacteria. Now the concept is that if after light illumination, it will interact with light, it interacts with oxygen, leading to ROS generation, which ROS can be further utilized for killing various cells, including cancer cell. So the disadvantage of this molecule is this molecule self-aggregates and very low soluble in aqueous system. So intention is to make this phthalocyanine available for the cell. So I thought of pegylation of this molecule. Now I chose copper because of its catalytic efficiency. Now, if we really want to mimic this GSH redox system inside the cell, what I have done, I have taken this molecule, I have taken added glutathione, which leads to sulfur-sulfur bond formation reaction to form GSSG. And here in this NMR, glutathione, when added this copper catalyst or the anti-cancer agent that I say actually. So it leads to within 5 minutes the glutathione oxidized form of glutathione formation. So I thought this will nicely work for as anti-cancer agent. So I have taken the concept is the copper phthalocyanine will form GSH to GSHG to make copper 2 to copper 1. Further, in presence of higher concentration of H2O2 in the cancer cell, it will generate ROS and undergo Fenton-like reaction for cancer cell death. This condition, the light is not required. Further, when light will be illuminated, this copper complex will interact with oxygen, will form ROS and will lead to cancer cell death. That's what we see. But PDT is detrimental for the normal cell as well. So if we really want to selectively kill cancer cell, we really have to go through this dark pathway. So what we did study with the normal cell type and A549, and we see for a normal cell when treated with this molecule without light illumination, this molecule, the cancer cell remain with 85% cell viability. However, with A549 you see the cancer cell death takes place even in absence of light, in presence of light. So I can say from my molecule can selectively kill cancer cell. Further, this FHD assay indicated that without light, this molecule still have some this oxidative stress. With light radiation, the oxidative stress increases. So that is because in the dark state where copper 2 interacting with GSH, 2 copper forming copper 1, that copper 1 undergo Fenton-like reaction leading to cancer cell death selectively. However, normal cell does not takes place. And that is specifically when I have done what kind of ROS is getting generated in when copper phthalocyanine were reacted with methylene blue in presence of H2O2, then leading to OH radical. You see in presence of methylene blue, this color changes to colorless, indicating OH radical is getting generated. So we can say that we selectively made a system which can kill cancer cells selectively where normal cells remain intact. So that's how I come to the conclusion. This is the highlight of my project. I presented these two work here, and this is my group. And yes, definitely I should acknowledge the contributor for this project is that my active collaborators are Professor S. Dr. Chaudhary in BRC, Professor Ganga Periyaswamy who does DFT for me, Professor Silpi Sharma who did biological study, and acknowledgement should go to BRNS, CNTTM for Smart Fabric, Anti-Cancer Application for CSIR, Enzyme Mimetic Study for UGCSRB. And I have this great group of students, and thank you very much for great listening. Thank you very much.
Question if anybody has? If no, then let's thank Dr. Navita.
I request Dr. Ramendra Pratap to felicitate Dr. Navita Sadukhan. Now I request Dr. Ravi Barnwal to felicitate the session chairs, Dr. Amrendra Kumar and Dr. Ramendra Pratap. Ravi. Now let's proceed with the next session, which is session 13. For that, I would like to invite the chairs, Dr. Ravi Barnwal and Dr. Mansi Roy.
So this is probably the last technical session, and there are three speakers in this session. So first speaker we have is Dr. Subash Chandra Ghosh, and he is a principal scientist at CSR Central Salt and Marine Chemical Research Institute in Bhavnagar. Dr. Ghosh did his PhD from IIT Kharagpur with Professor Amit Basak, followed by his postdoc at South Korea at NTU Singapore. He also spent some time in ICES A-star at Singapore as a faculty or scientist. He has been working in the field of catalysis, green chemistry, and sustainable chemical processes. He has guided multiple students, including 6 PhD students who are already currently working with him, and several master's students. And today he will be talking about the radioselective CH functionalization, annulation, and efficient route to medicinally relevant heterocycles. Dr. Ghosh.
Yeah, I'm already here. So, welcome all to the last technical sessions of this beautiful conference. I'd like to thank Jadhav sir and Dr. Vishnu Maya for giving me this opportunity to share our work, what we are doing right now. So, my topic on the radioselective switch functionalization and annulation reaction and efficient route to medicinally relevant heterocyclic synthesis and their functionalization. Yeah, sir, I will be very fast. So before going, I like to acknowledge my all supervisors. So I did PhD with Professor Amit Basak from IIT Kharagpur. Then I moved to South Korea as a postdoc in Posttech, followed by in NTU as a postdoc. Then I did job at Esther, ICS, and then from December 2012 onwards, I'm working as a scientist at CSR, CSMCRI. So my research topic mostly focused on CH activation and seed polysaccharides and synthesis of API and KSM. So today I will talk about CH activation and functionalization. So as I said, going very fast, actually yesterday Kamaljit also presented this one. So why CH activation people are working? So there are a couple of advantages over the traditional cross-coupling reactions. So the advantages is you don't have to use the pre-functionalized substrates. It is a step economical approaches and environmentally benign. The advantages is you can do the late stage functionalization. So when you have a molecule, you can do the late stage functionalization and you can synthesize the highly functionalized diverse molecular architecture. The advantage is the major challenge because you have a lot of CH bond. How could you specifically functionalize one CH bond in presence of others? So one such approach is to do the installation of a directing group. So directing group can be a translational directing group. It could be a you can reverse it, or you can removable it, or you cannot remove it. So directing group is nothing but a ligand. So it is an act as an internal ligand. So it coordinates with your metal and give that metal center close proximity to where which CH bond you can do the activations. So once it is activated, then you can do the functionalization. So if you consider simple benzene ring monofunctionalized, so there are 3 different hydrogen, ortho, meta, and para. But if you consider the naphthalene ring, so which has a one monofunctionalized, you have 7 different hydrogen. So how could you functionalize that? So if you have a directing group, so if it is a geometrically accessible center, so you can do the C2CH functionalization like here. So if you have a template assisted directing group like it a linker, then you can do at C3 position. if you change a little bit of directing group or the conditions, so that C8 position also can be functionalized. So that is also geometrically accessible. And another one, that interesting one, so which one is C4 position? So this one is not geometrically accessible positions. So here, what happens, the metal is chelated with, and then it goes to the single electron transfer pathway, and it generates a radical cationic species. So once it's a radical cationic species generated, that this positions, the C4 position is more reactive. It goes to the single electron pathway, so you can do the functionalization. So these are the major strategies which we followed in our reaction. The first one, okay. The first one reaction is the carboxylation reaction. So what we thought when we started this work, we checked the literature and we found that there is no such report on the carboxylation reaction. So we use CBr4 as a mask carboxylation reagent. So CBr4 can generate the CBr3 radical and it can do that reaction. So I'm not going in details of the reaction optimization. So after several screening, we found that copper triplet is a suitable catalyst, although catalyst loading is more, 20 mole percent, with potassium carbonate as a base. And you need to add DMAP also. And you can do the carboxylation at the C4 position of the naphthalene ring as well as in the quinoline ring. So this one, picolamide, is a directing group which acts as, which coordinate with the metal and activate that center. So we check the reaction mechanisms and all these things. So when we do the reaction kinetics, we found one interesting thing. We found one peak is coming and it going down. So that is an intermediate, and we isolate that intermediate. It is an orthoester, which we are expecting. So we did a lot of studies for the exploration of the reaction mechanism. So we did kinetic isotope effect and found that KH by KD is 1.04. So CH activation is not the determining step. And as expected, this goes through the radical pathway. So we trap both the CBr3 radical and this radical by BHT tempo. And we do the some other reactive substrate to know the reaction mechanism. We found that this, if you are N-methyl, there is no reaction. If you are, this is an ester, there is no reaction. If you change the pyridine to benzene, there is no reaction, and simple amine does not do the reaction. So that means the picolamide part is also important. And with that, all this data, we predict the reaction mechanism, like your substrate coordinate with the copper and goes to this pathway and goes to copper 2 to copper 3, CBr3 radical generate, then the ACT pathway, which one we are expected to form the radical cation, then the radical decombination and form this CBr3, and from the by the methanol methanolysis, you are getting the methyl ester. So in the substrate scope, well, it's not moving. Can you go to the next slide? Yeah. So okay, so then we did that. Then we did that amination reactions. So when we did the amination reaction with the similar catalytic systems with the copper catalyst, and we did the amination reaction at the C8 position, selectively at the C8 position. Okay, so I'm not going more details of these reactions. One important things we did, so we checked that we did the carboxylation reaction and we do the amination reaction. So we did the sequential reaction. First did the amination and followed by the carboxylation reaction. It goes well and giving the overall yield is 46%. And there we did the carboxylation reaction first and then followed by the amination reaction, and we got the similar kind of yield. So all these reactions can be scaled up very easily and with similar yield, and you can remove the directing group and reuse that one, and you can get the diamino product. So similar strategy, we use the sulfoxymination reaction. Sulfoxymination also one challenging reaction. Here also we did the same substrate with the sulfoxymidine and with cobalt catalyzed, and we got the sulfoxaminated product. Here also we explore all the possible pathway to explore the reaction mechanism like kinetic isotope effect, and in this case we found that for the cobalt catalysis kinetic isotope is 3.46. That means the reaction, that CH activation step is the rate determining state, although that hydrogen, this one, that it is not reversible. So we did the D2 exchange, deuterium exchange study, this is not this. And when we have some competitive experiment with the electronic, changing the electronic factor, we found that electron withdrawing group helps the reaction, so which might be the increase the basicity of this amine nitrogen, which coordinates with the metal, that gives our best result. And that also the true for the when we have a sulfoximazine with the methyl and bromo compound giving better results. So with that, we got this, and we this work is actually got the front cover page on the JOC, and we explored the substrate scope, and this also the scale up is very easy, and variation of several naphthylamines with different substrate, it works very well, and we did the variation with the sulfoxamazine also. This also works very well, and when we change the substrate scope with the analogous amide, this also works very well. So one challenge of this reaction when we were doing, we found that synthesis of a aryl alkyl ether, basically sterically hindered aryl ether, is very challenging. Okay, so two reports are there. Both are the decarboxylation approach. So you have a carboxylic acid with the electrochemical by Baran, and this one by Fu. They reported that photochemical one. So you generate this carbon radical and react it with a phenol, you can synthesize aryl alkyl ether. So when we are doing, so we thought that we are generating that copper complex. So if we make the TBHP, and if we can make the copper TBU, OTBU, this kind of complex, so then it can go through the single electron transfer pathway, and we can put the alkoxide, that is the bulky alkoxide, here in this position, C2 position. With this idea, we explored with the naphthyl substrate, and it really works well, and we found that it goes to the very good yield for all the naphthyl substrate. Not only the naphthyl substrate, with all the polycyclic amine, it goes very well. But when you have the two C2 positions, so there is some isomeric form. So this one is the major, which one is sterically less hindered, and minor product. So not only that, with the naphthalene or pyrene or anthracene system, it works well with the benzene systems also. And this one we are getting very good to excellent yield. So not only the TBAP, so we used this one. So with cumail peroxide, this also you can incorporate this kind of tertiary alkyl ether here. So we did the directing group removal, and this one you can synthesize 3 substituted benzene ring very easily. So with the copper and other systems, you can regenerally functionalize naphthalene at different positions like C2, C8, and C4 positions. So another work which one we are doing, the synthesis, regenerate synthesis, CH functionalization, and the annulation reaction. So you can synthesize that succinamide motifs is present in several bioactive moiety, and this kind of spirocyclic compound synthesis is very challenging. So starting from the simple precursors like an arene with an directing group and with melamide, you can do several reactions. One is the major one is the Michael type reaction, and you can do the CN spiroannulation reactions. You can do the CH-NH spiroannulation reactions, and there is no report which are doing this one with the CC spiroannulation reaction. So I'm going this one. So initially we did with the cobalt acetate as a catalyst, and with the eosin Y photocatalyst, we found that this reaction goes very smoothly, and we found the spirocyclic compound in excellent yield. And this reaction, as we use eosin Y photocatalyst, and we found that reaction substrate scope is huge. So with thiophene or naphthalene or benzene with lot of substrate, all the reaction goes very smoothly. And we check the reaction mechanism thoroughly, and we found that eosin Y, actually the role of eosin Y to modulate the cobalt 2 to cobalt 3, they are the electron transfer, and it makes this cobalt to cobalt 3, and it makes the activate the catalyst and goes through the CH activation pathway followed by the insertion, and then it goes through this pathway and giving the exocyclic moiety. So later we change the NN directing group to NO directing group, and we use our established catalytic condition, but it does not work. So we then again re-optimize the reactions and found that in the thermal conditions, cobalt acetate with sodium pivolite and silver as an oxidant, and this reaction goes well, and we got the spirocyclic compound. Next, we explored that whether we can do the same things with the acrylate. So when we did the acrylate chemistry, it does not work with cobalt. So we use the palladium catalyst with a copper as a Lewis acid actually. And then we explore the reactions, and it goes to the isoindolin, not the spirocyclic compound. This is isoindolin kind of moiety. And we apply these to synthesize some drug analog. But when we explored the reaction mechanism, we found that we have actually identified this kind of complex. So palladium and copper, the heterobimetallic complex actually is important for these reactions. So as with when we use the melamide, we are getting this kind of 5 member pyrocyclic compound, and here when we use the acrylate, we are getting this 5 member compound. So we thought if we use the naphthalene systems, and in presence of sub suitable acid, can we get this kind of reaction with a benzo quinoline systems with the pyrocyclic succinamides. So with this idea, we found that rhodium catalyst only activate these positions, and we got this all this kind of reaction, and these reactions also very good substrate scope. So we are yet to publish these results. We have filed this patent, and now we are exploring the reaction mechanism, and we predict that this goes through this rhodium goes through the first the combination, and then the carboxylic acid activate it, and followed by the CH activation, then the hydrometallation reaction, and the reductive elimination. Once this product is formed, which one we detected by ESIMS, and then the aldol kind of reactions, it goes to our desired product. So our last work is on the direct aldehyde CH functionalization reaction. So aldehyde CH functionalization is not so much reported. Only rhodium catalyst can do this reaction. So when we are doing this chemistry, we found that palladium catalyst can also do this reaction. So we isolated this complex, and we found that CH bond is activated. So when we did these reactions with aryl iodide, this reaction goes very well. And we established the reaction mechanism in this pathway. So we isolated this one, then the oxidative addition. But one such problem, we found that when you have an electron withdrawing group, then it does not work. Then we check the Hammett studies and all these things, and we found that if we use the diazo complex, which goes through the radical pathway, and it might works well for the electron withdrawing group. And as expected, when you use the diazo compound, the nitro compound, which is giving very good yield, and where you have a methoxy, which is electron rich arene, it gives the lower yield. So we established the reaction mechanism, and we found that this pathway goes through the radical pathway, and we trap that radical also. Again, we did these reactions with similar reactions with the photocatalyst. Here, actually, palladium TFA and a Janus ligand act as a catalyst, palladium catalyst, as well as it acts as a electron photocatalyst. So there we found that aryl boronic acid is reactive, and we can synthesize this all kind of ketone in excellent yield at room temperature. And we studied the other arylating source here, found that aryl iodide, which works in the thermal condition, it does not show good yield. And we did all these things to explore the reaction mechanism, and we have identified this kind of organometallic complex that you have a phosphine and phosphine oxide as a ligand, and we check the in situ phosphorus NMR, EPR, and all these things to establish the reaction mechanism. And reaction mechanism goes through this. The palladium activate here, and it forms this kind of organometallic complex, which we have identified, and form the acyl radical, which one we trapped by the radical inhibitor, and followed by these aryl boronic acid reactions, and we got the ketone compound. I think I'm in time. The final one is we did the similar reactions with aryl aldehyde, so with amine, and we got the amide in good to excellent yield for all these substrate, which works well. So and there we found that palladium catalyst, actually this one we have identified, and this one with the silver complex, this is the silver assisted acetate CH activation pathway, and we got this kind of complex when the amine reacted, and we got the amide product in good yield. So I am done. This is my group, and we are working. And thank you for all of you for your kind attention. I am free to take questions.
So thank you for finishing within the time. So maybe we can have one quick question.
Good afternoon, sir.
Yeah.
Sir, such a great work, sir. Sir, I have a question. In naphthalene core, you have functional at the C4 position.
Yeah.
So, sir, have you replaced that carbonyl with the sulfur bond? That carbonyl with CS bond? That is
Carbonyl with-
working with C or C4 functionalization or not?
Sorry, I'm not getting that. C4 functionalization?
That you have reported a C4 functionalization of
naphthalene, yeah.
Right.
Yeah.
So have you tried that amide? You are saying that amide is very good directing group.
Yeah.
So that carbonyl present, you have
Okay, no,
replaced
no,
that
no.
oxygen with
You are
sulfur.
saying as a thiamide.
Thiamide,
No,
right.
we have not tried that one.
Okay.
Okay.
Thank you, sir.
So, Amarend, I think he had questions.
Fantastic talk, very neat. I was interested.
Dihydrodium acetate.
The hydride intermediate that
Yeah,
goes
hydro-
through
yeah.
the
Yes.
cycle, you have invoked that dihydride intermediate is formed. Is it possible to detect it experimentally? It will be very interesting to see
Yeah,
that.
you can detect with NMR. NMR, the rhodium hydride peak, but unfortunately we are not getting. Okay, we have tried. Okay, because actually if you go through this, we are doing this one. So our solvent, sorry. So this one actually I have not mentioned. So we have actually adamantyl carboxylic acids. Okay. So that one actually prevent us to do this NMR studies. So we cannot make all CD adamantyl carboxylic acid to check the NMR, because when your NMR solvent is huge and it is only working with adamantyl carboxylic acid. So we are trying to do with acetic acid, means CD acetic acid, to get that kind of things. But until now,
It would
but
be very.
So we can have more question during the lunchtime.
okay. Okay. Yeah.
Thank you so much, sir. I request Dr. Ravi Burnwal to felicitate Dr. Subash Ghosh.
So we have a second speaker today. Dr. Dinesh Kumar. He is assistant professor at NIPER, Ahmedabad. Dr. Kumar did PhD from NIPER, Mohali with the Professor Asit Chakravarti, followed by his postdoc at NDSE with Professor Cook. And he has developed several strategies for complex synthetic challenges. And the title for his talk is a novel chemotype for oral cancer via dual CH bond activation relay protocol. So, Dr. Kumar.
Okay, so thank you very much. And I acknowledge Jadhav sirs and the organizing teams for inviting me. So I am going to discuss today is about the identification of novel chemotypes that resulted from the dual distal CH bond activation under palladium catalysis. So we know that in just like lectures that CH bond has a profound impact on the chemical society, ranging from the fine chemical to pharmaceutical. But obvious, it also has some challenges and limitations. So like a site selectivity, especially activation of distal CH bond, dual CH bond under the single catalytic systems, and a stereochemical outcome. For example, take the example. So like just take the example using the aryl alkyl quinazoline. So if you consider heteroatom as a directing center, there are many CH bond that could be functionalized. Also, is it possible to undergo like a delta CH bond activation under the catalytic system? And at the same time, whether can be have a two CH bond get activated under the similar catalytic system? So obvious people are working in this area using the several approach like steric and electronic biasness, template guided CH bond activations, enzyme inspired approach, and the photo reduction electrochemical method. So here I am going to discuss about the dual distal CH bond activation that we have recently published in the JICU. So we have thorough done the literature search and we found that there are 3 reports where the dual distal CH bond activation is reported, where at least one CH bond is distal in nature under the single substrate model. So if you see there like a activation of CH gamma, CH beta and gamma, and CH gamma gamma. Whereas our present study report the distal CH bond activation involving beta and delta CH bond activation. So it involves the alkyne annulation of aryl alkyl quinazolines with the alkynes to form a benzofulvene type of moiety, and this has been found a very good anti-cancer hit for oral cancer. So being a medicinal chemist, we are always in love with the heterocyclic compound. And this is not just a chance. If you see the recent 20 years, like all the FDA approved drug, especially a small molecule based drug, 85% are a heterocycles. And among these 85% heterocycles, at least 75% are n heterocycle in nature. And from this 75%, there are like a 60% is like anti-cancer drugs. So these are the top 30 heterocyclic system which are like reported in last 13 years in the FDA approved drug. And these are the like quinazolines, which are the like the basis of a study. It has a broad spectrum in case of like a biological activity. So we started reaction, the cyclization reaction of alkynes with the aryl alkyl quinazolines. If you theoretically possible, see there are multiple cyclization product is possible, which can be classified under the CH alkyne annulation and CH-NH alkyne annulation. So when we tried the reaction, we found that under the catalytic condition, we observed only one product, that is the 3A, and this formed the basis for further optimization and screening. So we have screened evaluated the different palladium and ligand system, and we found that under the palladium acetate and SIPR ligands, we are getting exclusively 3A with 72% yield. And this is the initially we thought that this is the structures. And in fact, we have wrote the CRG grant based on this structure and also be received. But when we took the single crystal structure, we found no, it is not the naphthalene, it is a benzofulvene type of compound which we are getting. So following the optimization study, we have done systematic substrate scope, and which is a very fantastic. If you see the functional group tolerance, it tolerates all the electron withdrawing, donating, halogens. In case of alkynes, also it works with the internal alkynes, whether it is symmetrical or unsymmetrical. However, the limitation is that it does not work with the terminal alkynes. So based following the substrate scope, we have done the few mechanistic investigation. So particularly we are interested in what are the other different kind of directing partner that can be used to direct this kind of distal CH bond activation. So we have done different pyridine, pyrimidine, benzothiazole, imidazoles derivative, and we found that these directing partners are non-productive under the reaction condition. Further, we protected the NH, we converted amide into thioamides, or with linear amide, it doesn't work. So that say that this quinazolones type moiety is very, very specific for this kind of transformation. Further, the question is that whether this cyclization proceed via intermediate asteroid quinazolone. So we monitored the reaction and we found that yes, we observed this asteroid quinazolone under the reaction system. However, when we put the reaction with preformed this asteroid quinazolones with the alkynes, it does not proceed the product. So it suggests that this is forming under the reaction condition, but it is not leading to the product. The product directly formed via the benzylic CH bond activation. As usual, we have done hydrogen deuterium leveling study, and also we have determined the Ki, kinetic isotopic effect, and we found that the distal CH bond activation, that is a delta CH bond activation, is a rate determining steps. So we propose the catalytic cycle. So initially we observe there is a formation of palladium-4 catalyst which undergo first CH bond activations with the benzylic hydrogens, followed by alkyne coordination and insertion to form the intermediate 4. Now, this intermediate 4 is very, very crucial. If this intermediate 4 undergo NH activation, it forms this kind of moiety, which is not operational under reaction condition. So it undergo the delta CH bond activation of the phenyl ring to form the corresponding benzofulvene, which undergo like a dehydrogenation reactions to form the desired 3A. So we have like a isolated this palladium-4 catalyst and thoroughly characterized, and we found this is the catalytic species which is a functional, and other isolated palladium catalyst is found non-functional under the reaction condition. So with this, once the synthesis is done, we next evaluated whether this type of compound we have any biological activity. Since being in Gujarat, we know that Gujarat is a hub for oral cancer. So we evaluated this compound for the oral cancer against oral cancer cell lines, Cal-29. So we found that the compound 4E, that is CF3 containing compound, is very promising for oral cancer with IC50 around the 9.41. And we have done the cell cycle analysis. We found that the compound 4E arrests the cells at the transition of S to G2 phase, and also it kills the cell through apoptotic pathway, not via necrotic pathway. We have further done the RNA sequencing and analysis, and we found that compound affecting the multiple hallmarks of the cancer, like it upregulate the genes which is responsible for suppression of cancer, and also it downregulate the gene for responsible for mitochondrial function like ATP generations. So considering all these biological activity, we found the compound 4E is very promising hit candidate for oral cancer. And so overall summary is that, so we report a dual distal CH bond activation, particularly delta CH bond activations to forms the noble benzofulvene type of compound, and it like override the 4+2 CH annulation, and also it does not follow the pedestrian phenylic CH bond activation pathway. And this compound is found very promising oral cancer hit candidate with having a negative effect on the cell migration, apoptotic, and the target multiple cancer hallmarks. So with this, I thanks again. And this is my research group. We are working in the area of method development, green chemistry, and two of my students also presented. I think one is presented, this is Garvita, presented the poster here, and one is the Sangeeta, is given the oral talk. So please, thank you very much. I am thank.
So in the interest of time, probably we can move to the next speaker, and you can all interact during the lunch time.
Thank you very much.
Good afternoon. We are about to end up our session, and today's last speaker, Dr. Joy Mitra, and she is currently working as principal scientist in inorganic materials and catalysis division at CSR Central Salt and Marine Chemical Research Institute. Her specialization is in supramolecular hydrogen bonded systems like hydrogels and metallogels for catalysis and electrocatalysis. She earned her PhD degree from IIT Kanpur in 2013 and also works on metal extraction and recovery from e-waste and spent catalysts to develop advanced inorganic materials. Today, she'll be delivering her talk about exploring amine-rich supramolecular silver metallogels for catalysis and as membrane targeting broad spectrum antibacterial agent. Ma'am, please. We are very much eager to hear you, ma'am.
Thank you. At the outset, I would like to thank the organizers especially Dr. Vishnu Maya, for this invitation and the opportunity to share some of our work with you. So as my title suggests, I will be talking about rich supramolecular silver metallogels and their applications in catalysis and some results of their use as antibacterial agents. Okay, so gels are viscoelastic soft materials where there are two coexisting phases, one solid phase and a bulk solvent phase. So what happens is the solid phase forms a 3D entangled network that can trap the solvent and immobilize their flow, thereby forming a gel. So as you can see from this figure here, out of these three images, only the C is a gel as it can hold its weight under gravity. And this is known as a tube inversion test and is the preliminary identification of the formation of a gel. So unlike polymer-based gels that was discussed by one previous speaker this morning, I will be talking about supramolecular or low molecular weight gelator-based systems, the formation of which is based on non-covalent interaction, and hence they are dynamic in nature. So these gels can be metastable and can crystallize or precipitate out during prolonged storage and is strongly solvent and counteranion dependent, especially in case of metallogels. So there's a prominent role of the counteranion, the solvent or the solvent mixture in formation or in governing whether it forms a gel or a sol or the material precipitates out. So the ligands that are used to form gels can or should dedicatedly have two types of centers, one that can participate in non-covalent interaction and the other that can coordinate with the metal center. Along with this, the property of the gels are strongly dependent on the property of the metal that has been used. So here are some of the common ligands that have been used in metallogelation. As you can see, mostly they are based on carboxylic acids or pyridine-based systems. The other heterocyclic moieties have been relatively less explored in this regard. Now, in view of this, we selected a small molecule, triazole with appended primary amine ligands as our gelating ligand. With this system, in a mixed solvent of water, methanol, DMF with water, THF water, and so on, we could form 3 gels based on silver, 2 from silver triplet, while 1 with a silver nitrate as the precursor. Now, all of these have a similar type of belt or rod-like morphology. Based on the molecular level characterization, we could see that in these species, silver exists in a silver 1 oxidation state. Also, in case of the nitrate gel, we could see that there are 3 distinct type of nitrogens that can be observed from the XPS analysis. Additionally, PXRD results suggest that the triplet gels are more crystalline as compared to that of the nitrate gel. Now, like was mentioned in the morning, so rheology is the most basic characterization of these gels. So we conducted rheological experiments under a fixed frequency by varying the strain percentage. And this showed that the nitrate gel was the strongest with the highest gel to salt transition temperature. Transition strain. So the reason behind was that the nitrate counteranion can form stronger hydrogen bond as compared to the triplet anion, which could be attributed, which could be a reason for its rigidity. Also, all these gels showed a frequency invariant behavior. Additionally, these gels were rheologically reversible or thixotropic in nature. By this, I mean that once a higher strain is applied, there's a transition from the gel to the sol state. And on removing the strain, the sol state can go back to the gel in a repeatable manner. So after the end of 5 cycles, all these gels recover more than 65% of their initial strain. Now, once we characterized that indeed we have a metallogel in our hands, we tried to explore the material for some catalytic applications. Literature survey revealed that in most cases, the supramolecular silver gels have been used as a precursor to form silver nanoparticles, which have then been used for hydrogenation or reduction reactions, either from aldehyde to alcohol or nitroarene reduction. But in our case, we wanted to use our material for some application that will take care of the prominent Lewis acidity of silver. Additionally, in our case, we also have the pendant amines, which can show some basicity in the system. So considering all these, in our search to find some reaction that will take care of the Lewis acidity of the silver, as well as the basicity of the amines, we chose the multi-component A3 coupling reactions for the synthesis of propergyl amines for this regard. Now, the reason behind choosing propergyl amines was that they are key intermediates in the synthesis of various nitrogen-containing biological active molecules, as well as in natural product chemistry. Now, this is the reaction of interest for us. This is a three-component coupling reaction of an aldehyde, an amine, and a terminal acetylene. So this is the product that we are interested in. But other than that, the diacetylene can also form as an undesired side product. So we checked the activity of all the three gels in this regard. We observed that both of the triplet, using both of the triplet gels, there was a prominent presence of this undesired acetylene dimer. But when we were using the nitrate-based gel, the amount of the acetylene dimer was reduced, and under heterogeneous conditions, it could be almost suppressed. Now, further, we moved along with this nitrate counteranion containing gel because that it minimized the steps by suppressing the dimer. Now, before we went for mechanistic analysis, we did for substrate scope, we did some mechanistic analysis. Deuterium labeling revealed that there was almost 100% deuterium incorporation. So the reaction went through a silver acetylide formation. Aside from that, a kinetic isotope labeling experiment revealed almost no effect, which showed that the rate limiting step is not this dissociation. We also used a radical trap to ascertain that the mechanism does not proceed through a radical pathway. Now, since we observed that the nitrate-containing counteranion showed a better selectivity for the desired propergyl amine product, we believe that this is how the mechanism is proceeding, and the nitrate anion, being a better hydrogen donor and a more symmetric anion in nature, also smaller in size, can bring the imine as well as the silver acetylide in closer proximity as compared to the triplet anion, which kind of favors the formation of the desired propergyl amine product. On the other hand, the higher bulk of the triplet anion sort of hinders this process, resulting in a higher amount of the acetylene dimer. Now, these are the substrate scopes that we have explored. We explored both aromatic as well as aliphatic aldehydes and observed that aliphatic aldehydes give a comparatively higher conversion under our protocol as compared to the aromatic aldehydes. Additionally, the amines were also varied, and we observed that 6-membered amines gave a better yield as compared to the 5-membered amines, and so on. Since this reaction was being done under a heterogeneous catalytic conditions, we also checked the recyclability of our protocol. So this reaction we could do till up to 5 cycles, and from there onwards, there was a slight decrease in the activity. but there was no silver leaching observed from ICP-MS analysis. What was happening is that due to the prolonged thermal reaction condition, some part of the silver one was being reduced to silver nanoparticles, which decreased the yield of the product. Now, switching gears, I will be discussing another example of silver-based metallogels as membrane targeting broad spectrum antibiotics. Now, antimicrobial resistance is a big problem nowadays, and it is probably going to increase in the coming years due to the extensive use of traditional treatment approaches. To counter this antimicrobial resistance, membrane targeting compounds are being explored recently, but they have mostly been limited to amphiphilic systems and also synthetic mimics of antimicrobial peptides. Now, we wanted to explore the silver-based hydrogels that we synthesize in our lab. The reason behind using these hydrogels is that they are in general biocompatible with a high water content. They can cater to sustainable release of drug or other payloads inside it. Supramolecular gels are easy to synthesize and functionalize. And in our system, since there were pendant primary amines, we wanted to explore the hydrogen bonding opportunities and their membrane targeting potentials. Also, a literature survey revealed that triazole and urea-based systems have been known to be active against escape pathogens. Now, with all this in mind, moving on from our previous gels containing silver and the triazole species, we added a small amount of urea into the solution to give rise to two multi-component silver gels where there's both the triazole and urea as the gelatin ligand. Now, in this regard, we explored two bacteria, the gram-positive Staphylococcus aureus and the gram-negative Campylobacter jejuni. Both of them showed an MIC 50 value of around 60 micrograms per ml. In addition to testing their efficacy against gram-positive and gram-negative bacteria, we also did some cytotoxicity assay using various cell lines. And our material showed mild cytotoxicity, which was distinctly less than the individual components that constituted the gel. Now, one of our goals was to check their membrane targeting potential. In this regard, SEM images at different loadings of our material showed that with increase of loading, there's a prominent disruption in the bacterial cell membrane, as has been showed by the red arrows here. In case of both the gram-negative Campylobacter as well as the gram-positive Staphylococcus, as you can see from here. Now, we believe that along with the hydrogen bonding interaction that can interact with the negatively charged phospholipid membrane of these bacterial systems, the positive zeta potential of the silver hydrogels is further facilitating the electrostatic interaction with the negatively charged membrane, and the cumulative effect of the hydrogen bonding as well as the electrostatic interaction helps in the disruption of the bacterial membranes. So other than bacterial cell membrane disruption, we are also preliminarily checking the chances of reactive oxygen species generation in these materials. So for both the bacterial cells, we did some microscopic analysis by incubating with DAPI as well as PI stains. Now, why we use these two stains was that the DAPI stain can stain the nucleus of both the living and the dead cells, while the PI can only penetrate the cell once there's a membrane disruption and can identify the dead bacteria. So by both these staining, we observed a prominent ROS generation in the system, which was further quantified by time-dependent experiments, which showed that the ROS generation is maximum in about 1 hour, but about 2 to 3 hours, the ROS generation, though decreasing, is still maintained, giving rise to a sustainable ROS generation over time. Now, to summarize this, In this case, we have observed the cumulative effect of terminal amine-mediated hydrogen bonding interaction, positive surface potential, which helps in disrupting the phospholipid membranes. And this was coupled with ROS generation, overall resulting in the bacterial cell death. Now, summarizing what I've discussed today is that we talked about silver diaminotriazole-based metallogels that have been used as a heterogeneous catalyst for F3 coupling reaction to form propyl and multi-component gel from silver along with urea was synthesized, and this showed efficacy against both gram-negative and gram-positive bacteria. Now, before I end, I would like to acknowledge my students who did some of the work here. Financial support from my parent institute as well as ACRB Power. And thank you all for listening.
Thank you, ma'am, for your very much exciting talk. We can have a few questions if you have any question. The question answer session is open now. No question. So we can move on to the other sessions. Alicia.
Thank you so much, ma'am. I request Dr. Manasi Roy to felicitate Dr. Joy Mitra. I request Dr. Rupesh Maurya to felicitate the chairs of this session, Dr. Ravi Barnwal and Dr. Manasi Roy. The past 3 days have been filled with enlightening discussions, innovative research presentations, and valuable networking opportunities. Ladies and gentlemen, we have now reached the final session of our conference, the valedictory session. For that, I would like to invite Dr. Karthik Jain, Registrar and Patron of SIDD 2025, for his valedictory remarks.
So good afternoon, everyone. Respected Dr. J.S. Yadav, sir, all the deans of his school, Dean Research, my dear faculty members, students, and participants. As we gather here today to mark the conclusion of the first international conference on sustainable innovation in drug development, I stand before you filled with a deep sense of gratitude, inspiration, and hope. Over the past few days, we have witnessed groundbreaking discussions, remarkable research, and a shared commitment to a future where drug development is not only innovative but also sustainable. This conference has been more than just a meeting of minds. It has been a celebration of human ingenuity and dedication. Each one of you has played a vital role in shaping the future of medicine, pushing the boundaries of scientific exploration while keeping sustainability at the core of your work. Your ideas, your collaboration, your relentless pursuit of knowledge will define the next era of pharmaceutical advancements. As we conclude, let us remember that this is not the end, but a new beginning. The insight we have gained here must now be transformed into actions. The world looks up to us to innovate responsibly to create affordable and accessible drugs. and to ensure that scientific progress all of humanity while preserving the delicate balance of our environment. To the young researchers and students among us, your passion is the driving force of this industry. Let this conference be the stepping stone in your journey. To the experienced professionals and pioneers, your guidance, wisdom continue to inspire the next generation. Your contribution to the field serve as beacon of hope, reminding us that great progress is made when knowledge meets perseverance. Thank you all for being a part of this momentous journey. May your research thrive, may your ideas flourish, and may we meet again to celebrate even greater milestone. So like this tradition has been started by Indishil University, where we'll be doing signature international conferences in a specific month. So let's hope we'll be meeting again in the same month, but next year. Thank you all.
Thank you so much, sir. Now I would like to invite Dr. Vishnumaya Besai, convener of SIDD 2025, for her concluding remarks.
Good afternoon, everyone. Respected Dr. J.S. Yadav, sir, respected Registrar, sir, Dr. Karthik Jain, all deans of the School of Science, School of Pharmacy, and School of Engineering, Dean RNI, all administrative staff and participants, and my dear students. So already our registrar sir has more or less concluded this conference. But on behalf of organizing committee and as a convener of SIDD 2025, that is Sustainable Innovations in Drug Development, I would like to conclude in quick 2-3 lines. That is, when we hold such kind of conferences, the idea is to bring on the same platform what is our aim, people who can discuss our aim and the outcome we want it should come out. So the aim was to bring the experts from different fields in the area of drug development, and starting from the keynote lecture by Padma Shri Professor V.K. Singh on the title like creativity in synthesis and how it can be used in the area of drug development. Then plenary lectures, which was completely filled with interdisciplinary approaches by Professor G. Muges, ISC Bangalore, and Professor Tapas Kundu, JNCSR, and then followed by other exemplary experts from industry as well as academia. And then we had a lot of series of experts for invited lectures, and our students, I am sure that they got huge opportunity to discuss and go through all these sustainable practices which is going on currently in the area of drug development, and I am sure that they got benefited. And most importantly, the highlight of this conference was a student interactive session, and we found that the experts, they gave a very elaborate explanation, and as well as they answered our questions put through from the student side. And we are very happy from the organizers side, especially we are thankful to our chief patrons, Professor Dr. DJ Shah, our provost, and Dr. J.S. Yadav sir, for giving us this opportunity to hold this conference. And let us cheer up in the name of sustainable practices. We wish you all the best. And now I give it to the our next who is this? I would like to invite Dr. Polumi Sengupta, the member of organizing committee, to proceed with the announcement of awards and certificates. Thank you so much.
Good afternoon.
Audible. We are waiting for the list. Okay. First, we will announce the poster awards. So we will start with the RSC poster awards, the awards which have been sponsored by Royal Society of Chemistry. For distributing the awards, I would like to invite registered Dr. Karthik Jain sir and research director Dr. J.S. Yadav sir on the stage, please. I invite Dr. K.N. Tiwari, Dean SOS, Dr. Vijay Singh, Dean RNI, to do the honor. Shali and Vishnumaya, please. So for the first prize, okay. So these are all equivalent prizes, not first, second, third. I invite Shipra Somani from IIT Bombay. For one of the RSC special prizes. No rank. Yes. Congratulations. Thank you. The next person is Miss Mohini Shahaji Ghorpade, IIT Gandhinagar. Next name is Gori Chandani Suresh Bhai, Saurashtra University.
Okay, she is awarded in the name of Green Chemistry Journal from RSC.
Next is Shyam Nilesh Bhai Sadhu. He is Gujarat University. Is in the name of chemical communications. The last RSC sponsored prize is for Rupali Sudhir Chaudhary, Gujarat University. It's in the name of RSC Advances.
No, one thing is that there few of them are MSc students as well, and these poster prizes are accompanied by cash prizes as well. Yeah.
Yes, please wait for the cash prizes as well.
So online transaction.
So we will start with the ACA sponsored special poster prizes. The first name is Jyoti Sai Phanindra Kumar, IIT Gandhinagar. Next one, Amardeep Singh, Nirma University. Amardeep Singh, Nirma University. Patel Nirab Kumar Prahlad Bhai, Indrashil University. It's sponsored by Organic Letters. ACS organic letters. The last ACS sponsored prize goes to Chaudhary Hiren Kumar Ramesh Bhai from Sakalchand University. Sponsored by JOC. So with this, we complete the poster prizes. We will move to oral presentation prizes. The first name is, okay, these prizes are sponsored by RSC, Royal Society of Chemistry. The first prize, like one of the RSC sponsored prize, is Miss Dharti Mistry, Central University of Gujarat. Hmm. Yes, yes, please come. Next is Kinnari Janardhan Shukla from IIT Gandhinagar. This one is sponsored by Chemical Science. Next, Shinde Sangeeta Dattatre, Naipur, Ahmedabad. This is sponsored by Green Chemistry. She's coming. She's coming. Next is Miss Juhi Pal, IIT Bombay. Juhi Pal, IIT Bombay. Organic and biomolecular chemistry. Next, Mr. Shekhar Kumar, IIT Gandhinagar. RSC Advances sponsored prize. The last one in the offline oral presentation is Pandya Vikrant, Indrashil University. This is sponsored by Indian Photobiology Society. We are starting online oral presentations. I wish there is any recipient. Let us announce the names. These prizes are sponsored by IPS, that is Indian Photobiology Society. The winners are Akshay Nathubhai Bharodia from SVNIT. Next is Zaheen Akhtar, CSIR IIM, Canal Road, Jammu. Suraj Das, ISAR Bhopal. Trayambek Nath Chobe, BHU. These all are sponsored by IPS. There is a correction. One of the ACS sponsored prizes, we announced Amardeep Singh from Nirma University. He's actually from Naipad. So if there is any person from Naipad, they can please come. Oh, you are there. Okay, Clark. No, no, he's there. Yes. Yes. Amardeep Singh, one of the ACS sponsored prizes. Yes. This is organometallic sponsored. So all the certificate recipients, please know that cash prizes are involved with it. So make sure you connect with Vishnu Maya madam, who is the convener of the conference, and you get the cash prize, sign the receipt and leave. Before leaving, you should sign the receipt. Okay, we are moving to the next session, that is delivery of good wish message by Dr. J.S. Yadav, sir, please.
Dear all, I'm not going to name everybody. I'll name only one or two at the end of this. I think this 2.5 days, you had a good feast of chemistry, which was covered like asymmetric synthesis, medicinal chemistry, organometallic, CH activation, green chemistry, almost everything. And also bioevaluation of some of the compounds, inorganic, bioorganic, all these topics are covered, and they were all given by the expert from the discipline, because you have chosen the expert very carefully, so that all kind of knowledge you gain when you go back home. The other thing is, I hope you people have interacted with some of the people who are working not exactly in your area, like medicinal chemistry people should have interacted with the biological evaluation people, vice versa. Or green chemistry people would have interacted with the industrial thing, so that you have opportunity to collaborate with this. And you can, once you collaborate, see now the even the funding agency wants that you people collaborate, different institute, different people, so that funding can be given. From the individual perspective, that is not preferred nowadays. So they prefer teamwork, collaboration. So I hope you may make a note. I hope you have developed some collaborator with these 2.5 days. So, and if you're not, you can communicate. I'm sure all people's email ID will be available. So when you, I'm sure when you go back home, you have got quite good knowledge, and I think our registrar has nicely prepared and given the lecture. So I'm sure some of the things you will remember, and I'm sure you do that. What I can say is, when you conduct this kind of successful confirm, that you people have to decide. But there are some people's whose contribution cannot be denied or ignored. I'll say one of these people which have got comfort, table transport without fail, food, lunch, dinner, everything was done by a single-handed people by Dr. our Rupesh Maurya. Where is Dr. Rupesh? थोड़ा खड़ा हो जाओ देखो. You know, whenever I was getting up in the middle of the night, he's saying he's there. 5 o'clock is there, 4 o'clock is there, 11 o'clock is there. I ask him whether you are finding time to sleep. Sir, aa raha hu, ja raha hu. So that way he has done. For last 3 days, I'm thinking you'll get enough rest now. 3 days he has been awake. Sunday, Saturday, Sunday, Monday, Tuesday. Today is Wednesday. Today I think you will get a good sleep. And another, of course, Vishnu Maya is convener, so she doesn't need to be thanked. But definitely, all of you can appreciate the kind of efforts she has put. And along with, I, I, I will be failing in my duty if I don't say, along with Rupesh Maurya, you just name them who are the people, because I can probably such as notice Sachin and the Kalpesh, but there are more people who have helped you. Huh? Let them stand at least, if they are there. Sachin, Kalpesh, Nirav. See, these are the people who are made your transport, stay, food, everything comfortable. So they need more clapping, okay? It was done under his guidance.
All the PhD students are requested to please come on stage. All the PhD students are requested to please come on the stage. You all have worked so hard.
Come on. Hardik.
And all the volunteers, BSc and MSc, please come on the stage. Volunteers. अरे सब आगे बैठो आओ ना सब आओ अनाउंस करो आ जाओ अब आ जाओ विष्णु माया भीड़ बहुत ज्यादा है हाँ तो नीचे बैठ जाओ आ जाओ दोनों आ जाओ आओ जल्दी आओ आ जाओ नहीं करना भी जल्दी जल्दी आओ मान से ना one day to give these people Thanksgiving आ जाओ आगे बैठ you have also worked behind the scenes. Please not acknowledge, I know. Come आ जाओ आइए हल्के सर आइए प्लीज प्लीज आइए सर अरे प्लीज सर आइए मेमोरेबल आपसे कुछ तो कुछ बीच में पूछते ही रहिए इस साइड में खड़ा नहीं प्लीज आप इसमें आइए नहीं बीच में नहीं इधर आप अंदर जाइए ना आप अंदर जाइए अरे अंदर जाइए आप मैं इस साइड में that is like I'm grateful but हम तो ऐसे नेचुरली पूछ लेते हैं तो थैंक यू सर Thank you. Thank you.
Okay, I think all of you are delayed quite late for lunch. Only thing I can say that please. अभी अभी नहीं मिस मैं बोलता हूँ चलो So that was from the scientific side. And other is our registrar Praveen, Harshit Bhai, पता नहीं किधर है वो है, Harshit Bhai, finance, and Praveen all management, administrative management he has done. Then we have our registrar, our Nimish Bhai. There are so many. If I keep on giving name, there are many people. But leader of all was our Praveen Bhai, and assisted by Harshit Bhai, who was the financial. अरे विजय की जरूरत नहीं है. These are all non-science people. Okay. विजय को विजय नहीं, हम बस विजय all scientists automatically get credit.
थोड़ा आगे ले जाओ प्रोजेक्टर ओके ओके फाइन
Science and administration goes together.
Yes.
Now you know why the conference was good, because convener never listened to me, okay? Okay, we all done. Only last thing I can tell you, if you feel that it is very good, convey to all other people that it was you attended and it was very good. And I thought, Vishnu, I thought our faculty was also done the volunteer, no? A lot of volunteering.
Yes, has those names. Yes, sir, our faculty have volunteered.
I think other member also have done, I'm sure. We'll have one. Sir, क्या उनको भी बुला दो? Who are there? अच्छा, वो तो हाँ हाँ अच्छा.
On behalf of Sir, I would like to name few of all the SOS faculty members who have contributed to this organization, organizing this conference. So starting from Dr. Rupesh Maurya again, and then Dr. Ankit Srivastava sir, and then Dr. Alka Shankar, Dr. Ritu Patel, Dr. Sanjay Khan, Mr. Hemendra Tang, Dr. Manasri Roy. I request all whoever I name, please come over the stage. Dr. Haren Giri Gosai. Haren sir. Dr. Polomi Sen Gupta. Dr. Manas Pal. Sanjay sir, please. And anyone, and one very crucial name is Sadanandji also, sir. Here. Yeah, he is there. He is there. Dr. Dargaya, sir. Dr. Randeep Kumar, sir. And one very important invited speaker as well as our faculty member, Dr. Nidhi Gaur. Please come on the stage. Kindly excuse me if I missed any name. Any SOS faculty member, if I am missing any name, please come over the stage. Yeah. Okay. So, our heartfelt gratitude to one and all. Thank you so much.
आ जाएगा.
Only thing I'll say that if people have to find somewhere, start coming anything, you write to her, write to our convener, so that we can have improvement next time, maybe copy to me. And also, if you liked it, also tell to others that it was a good conference or anything about our campus, about our greenery, about the conference, anything. Okay. So that's what I'll see. Now we'll ask what is your name? Saidi Rana to propose vote of thank. हाँ? तो आप ही करोगे ना? का नाम
I think Yadav sir conclude everything in a vote of thanks, but I would like to give special acknowledgement to our funding agency, uh, Kerala Pharmaceutical, DST, RSC, ACS, IPS, Indian Photobiological Society, Broker, CSIR, Guj Coast, and Aditya Scientific Industry, GSK Chemical, and Holy Scientific sponsor. Actually, without them, it's very difficult to arrange this big conference. So I will give a special thanks to sponsor. I will give special thanks to our sponsors and IT team member Sudeep, Harsh, and Hitesh sir. Your technical expertise ensured that our session ran seamlessly, enabling a smooth virtual and in-person interaction. And I think Yadav sir gave all the vote of thanks to all the committee member. Now I would like to say, everyone, please stand up on their place for the national anthem.
अब हम राष्ट्रगान शुरू करेंगे। जनगण मन अधिनायक जय हे भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविण उत्कल बंगा, विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा, तब शुभ नामे जागे, तब शुभ आशीष मांगे, गाहे तब जय गाथा, जनगण मंगलदायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे। भारत माता की, भारत माता की, भारत माता की। धन्यवाद। With this Rashtragan, this conference is declared closed. Thank you so much. And please enjoy your lunch, which is kept near the faculty residence lawn. And just a quick 1 minute. We are going to have another event in our Indrashil University. For that, I would like to request Mr. Hemendra Tang to quickly announce that. So on behalf of that, our one of the student from forensics, Ms. Suchi Smita Patra, she will announce that.
Good afternoon, everyone. It's a great from Department of Forensic Science, School of Science, Industrial University. We are organizing a one-day workshop for hands-on training for instrumentation techniques used in forensic analysis on 9th of April from 9 a.m. to 4 p.m. And it will be chaired by Dr. Vinay Kumar Srivastava, who is a highly decorated security trainer with experience in 100 plus post blast investigations and intelligence and his expertise will help us understand how scientific techniques play a crucial role in real life crime investigation. And this is an offline event, and the registration mode is hybrid. The fee is 399 only, which includes the kid breakfast and lunch. So if you all are passionate about forensic science and eager to apply, so please don't miss this opportunity. Thank you for your time.